Yang YouTube-nya masih sama kah, Mbak? Beda pasti ya.
Tidak sekarang aku.
Kamu acara BEC jam berapa, Mbak?
Mulai jam 8, tapi speakernya ntar jam 9.
Oh,
Karena di sini kelar dari
oke.
jam. Kita jalan.
Wait, aku cek YouTube-nya ya. Ada sih, masih.
Wah, kita jalan sih di aku ini.
Kenapa?
Ini udah jalan. Oke.
Iya, oke. Rahma boleh dicek slide-nya, mungkin slide waiting-nya ya. Ini udah ada yang mulai masuk. Bentar. Kita buka jam 8 aja ya, teman-teman.
Jam 8. Kita jam 8 aja.
Oke.
Kan saya izin keluar dulu ya. Mau jalan ke kampus.
Oh iya, Mbak. Oke.
Nah, terima
Makasih,
kasih.
Mbak Dini. Ya.
Oke. Bu Daniar, nanti saya ada ini ya Bu, kayak Hello Miss Daniar, how are you today? Terus are you ready to lead us to the main event and the Q&A session? Jadi kayak eh apa yang menyapa Ibu dulu gitu buat siap nggak buat memberikan materi gitu ya Bu, memandu acara gitu ya Bu.
Oke, oke. Ini kita mulai admit jam 8 aja ya, teman-teman kali ya.
Iya Ibu.
Oke.
Kalau suaranya begini lebih jelas nggak Bu?
Coba di kata-kata ada Mas kamu MC. Coba Din. Halo? Halo?
Oh iya, iya Bu.
Iya.
Ternyata masih di mute. Oh ya, coba ya Bu.
Ya,
With the introduction conclude, I'm delighted to introduce our moderator, Miss Daniel Fiorini. Jelas nggak Bu?
jelas tapi memang agak kurang jelas dibandingkan kalau kamu ngomong bahasa Indonesia ya.
Oh, iya Ibu.
Oke, oke, jelas sih. Masih oke lah.
Oke.
Ini di admit aja apa ya? Udah banyak teman-teman. Saya admit aja ya.
Assalamualaikum Bu Daniel.
Waalaikumsalam, Bapak.
Rencana jam berapa nanti?
Kalau di rundown-nya mulainya 8:30 sih, Pak.
Setengah sembilan itu
Ngumpulin.
sambutan Pak MC dengan Pak Ismar ya.
Iya, Pak, betul.
Oke ya. Assalamualaikum Pak Ismar. Good morning, Mrs. Jaspreet.
Waalaikumsalam, Pak Rusdi.
Ya,
Good morning.
Good morning, Dr. Jasper.
yeah.
Good
How
morning.
are you today?
I'm good. How are you?
Ya.
Ya,
Mohon izin nanti Pak Ismar yang membuka ya.
ya, boleh, Pak.
Oke, saya ikut aja silakan. Terima kasih.
Ya,
Thank you, Dr. Jaspreet, for your coming in our general lecturer.
Thanks for
Yeah.
inviting me.
Yeah.
So Dr. Jasper, join with us online or offline?
Online.
Yeah.
Online, Pak Ismar. Yeah.
Yeah, I'm joining you online. Yes.
Yeah. So Dr. Jasper, what time now in India? Or
It's
oh,
10 minutes to 7. It's 7
10
a.m.
minutes to 7. All right.
Yes.
Yeah. So we had time different about 1 hour. 1 hour, yeah. Right.
Yes, 1.5 hours.
Yeah.
Yes, you're ahead by 1.5 hours.
Mhm. Yeah.
Hi,
Good
Dr.
morning.
Jasper. Hello, Dr. Daniel. How are you? Nice to meet you. Fine. Thank you, Doctor, for coming in our event today. Thank you so much. As I said, thanks for inviting, and hopefully I can convey up something to the students. Yes, of course. Thank you.
So
Thank
if
you,
I understand, the students are mainly from biological background, right? They do not have a lot of bioinformatics related practical experience.
Yeah, our student major is biology education, doctor. So, but they need the content about bioinformatics, of course.
Okay.
Terima kasih Pak Ismar sudah menyempatkan
Yeah.
Bapak.
Nice to meet you, Dr. Dania.
I'm not doctor, Pak Ismar. Thank you.
Yeah, you're welcome. Thank you very much.
We're still waiting for the participant, Dr. Jaspreet. So maybe we will start at 8:30 in Indonesian time. Yeah.
Yes. Sure.
Recording Bu Daniar, di-record.
Oh ya, sudah di YouTube sih Pak, foto ke di YouTube.
Oke, ya.
So the participant is student and also lecturer. The participant.
Iya Pak, lecturer and students. So, Dr. Jasper, maybe Pak Asmar is our what we call Pak Vice Dean, Vice Dean for Academic Affairs from Faculty of Mathematics and Natural Science in Universitas Negeri Jakarta.
Yeah.
Nice to meet you, sir.
Also nice to meet you, Dr. Jasper. Thank you very much for joining us.
Welcome, Dr. Jaspreet. Welcome all participants to our present. Before the event starts, all participants that include all of the students and perhaps the lecturer here are expected to adhere to the following rules. 1. Participants are expected to their full name. 2. Participants are expected to turn on your camera feature and mute the microphone before being allowed by the moderator. 3. Participants are expected to use the virtual background provided by the committee. 4. Participants are expected to fill in the attendance form using the link provided in the room chat. Good morning and warm greetings to all of us. Honorable Mr. Dr. Esmar Budi MSc, Vice Dean 1 of Academic Affairs, Mr. Dr. Rusdi M. Biomed, Coordinator of the Biology Education Study Program, Ms. Dr. Dahlia Sukmawati, MSc, Coordinator of the Biology Study Program, Ms. Dr. Supriyatin, MSc, Coordinator of the Master of Biology Education Study Program, respected faculty members from the Biology Education Department who have graciously joined us, my esteemed fellow participants, we also extend our heartfelt appreciation to our distinguished speaker, Dr. Jaspreet Dhanjal, Assistant Professor in the Department of Computational Biology at the Indian Institute of Technology, Delhi, for generously sharing her expertise with us today. We begin by expressing our gratitude to God for His blessing and the hope that allows us to gather here for today's visiting lecture session. We are excited to continue our series of public lecture and guest lecture this October titled Innovation and Development of Bioinformatics and Their Role in Education. To start, allow me to introduce myself. I am Inin Rahmawati, a biology education student from Universitas Negeri Jakarta, class of 2022, and I'm honored to serve as your master of ceremonies for today's event. Our agenda today includes 1. Recitation of prayers, 2. Singing the national anthem Indonesia Raya, 3. Opening remarks by Dr. Esmar Budi MT, Vice Dean 1 of Academic Affairs, 4. Introduction by our moderator, 5. Speaker introduction, 6. Presentation by Dr. Jaspreet Dhanjal, 7. A discussion session, and finally the closing. Before we proceed, let us begin with a prayer. I invite everyone to take a moment to pray for the smooth proceeding, protection, and valuable knowledge acquisition for all participants. Time and place, the prayer begins. Thank you to all. May those who prayed be rewarded with goodness. Amin. Ya Rabbal Alamin. Moving on, we're singing the national anthem Indonesia Raya. Please sit in a good position. To invite Dr. Esma, Vice President of Academic Affairs, to deliver the opening speech. The Dr. Esma Budi, MT, the floor is yours.
Yeah, thank you very much to Ananda Dini Nurahmawati. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning to all participants. The Honorable Dr. Rusdi and Biomed as the coordinator of the Biology Education Study Programs. The Honorable also the Dr. Dalia Sukmawati as the coordinator of the Biology Study Program, and also Dr. Hanum as the coordinator of the Magister of Biology Education, and of course the Honorable of our special visiting lecture today, Dr. Jaspreet Khaur Dhanjal from the Department of the Computational Biology, Indra Pasta Institute and Information Technology Delhi, and also the Honorable all the lecturer, students, and also all the participants. Alhamdulillahirrahmanirrahim. To Allah Subhanahu wa Ta'ala. So today we all gather here in the seminar of the visiting lecturer with the theme of the innovation and development of the bioinformatics and their role in education. So first of all, I would like to greet Dr. Jaspreet. Thank you very much for your join us and join us in our department and our faculty, and also in our university. Hopefully, in the future, you also welcome us offline. Thank you very much to join us in seminar with the theme of the innovation and development of the bioinformatics and the role in education. So Dr. Jaspreet will share the information or knowledge or skill in how the bioinformatics implement in the biology research and also in biology education. So Bapak Ibu, as we know that bioinformatics is the multidiscipline that combine between the biology science with the technology information and also computer science. So nowadays we also know that the bioinformatics is utilized, for example, in analyze how to analyze the data. Yeah. And also it can be also implement in the education and also in teaching, especially in the biology field. So in this occasional, I would like to thanks to Dr. Japet and also to all the lecturer and also to the participant that join in this seminar that we can discuss sharing our information, knowledge, and also skill, especially in the bioinformatics for the biology research and also for biology education. Baik, allow me to declare in bahasa Dr. Japet. So Bapak Ibu, tentu sama-sama kita ketahui bahwa bioinformatika ini adalah bidang multidisiplin yang menggabungkan antara biologi dengan ilmu komputer dan juga teknologi informasi. Dan seperti kita tahu sama-sama saat ini tentu kedua bidang tersebut perkembangannya sangat cepat sekali sehingga memang mau tidak mau kita memberikan dampak pada pengembangan bidang ilmu yang saat ini kita geluti, khususnya tentu di bidang biologi. dan tentu juga akan berdampak pada bagaimana kita memberikan pengajaran ataupun pembelajaran, khususnya di pengajaran dan pembelajaran biologi. Insya Allah dalam kesempatan kali ini kita bisa berdiskusi, saling belajar dan juga saling memberikan pengalaman terkait dengan bagaimana penerapan bioinformatika dalam riset, dalam penelitian dan juga dalam pengajaran atau pendidikan. Terima kasih untuk thank you for all, especially for the biology education physics program, Dr. Rusdi and Biomed, and also Ibu Dania, and also to all the lecturer that has facilitated of this seminar. That's the speed, and on behalf of the Dean Faculty of Mathematics and Science Universitas Negeri Jakarta, with saying Basmalah, Bismillahirrahmanirrahim, the seminar is officially open. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Esmar Budi MT, for your insightful remarks. We will have a documentation session to capture this memorable moment. I kindly request everyone to turn on your cameras as we take a group photo together.
Dini boleh diaba-aba mungkin, Din.
Iya, Ibu. On my count. 1, 2, 3, cheese. Again. 1, 2, 3, cheese. Okay. Participants are expected to fill in the attendance form using the link provided in the room chat. Okay, moving on. With the introduction concluded, I'm delighted to introduce our moderator, Ms. Daniar Setiorini, MPD. Hi, here Ms. Daniar Setiorini is a lecturer biology education from from 2019 until now. His research article include augmented reality (AR) technology on the Android operating system in human respiratory system from organ to cell. Okay, hello Miss Daniar.
Hello.
How are you today?
Okay, good. Thank you. How about you, Dini?
Okay, I'm good. Miss Daniar, are you ready to lead us through the main event and the Q&A session?
Yes, of course. Fine.
Okay. I kindly request to that all participants turn on your cameras to Miss Daniar Setiorini, MPD. The floor is yours.
Okay, thank you, Dini, for your wonderful introductions. Yeah. Yeah. Right. So we will begin the main agenda for today biology education visiting lecture. So maybe I will open first for this session. Assalamualaikum warahmatullahi wabarakatuh. Good morning to everyone. Honorables, Bapak Dr. Eksmar Budi MT, as Vice Dean of Academic Affairs, Faculty Mathematics and Natural Science, Universitas Negeri Jakarta. The Honorables Dr. Rusdi M. Biomed as the head of, sorry, coordinator of program education, biology education, Universitas Negeri Jakarta, and all the lecture and participants who come in this event today. Welcome to today Biology Education Visiting Lecture 2024 on Innovation and Development of Bioinformatics and the Role in Education. My name is Daniar and I will be your moderator for this session. We have an exciting topic and session that will inspire and engage your thoughts. And first, I would like to say thank you to Associate Professor Dr. Jaspreet Khaur Dhanjal as our speaker for today. It is really an honor to have you in here with us, Doctor. I'm really excited to hear your lecture sessions today. Okay, before we begin, I'd like to inform you the detail of this session, sorry. First, speaker will present the material for about 90 minutes. After that, there will be Q&A session for 15 minutes per session. Maybe we will have 2 or 3 session or termin for Q&A session. If you have any question, I will give the opportunity to participant to ask question directly to our speakers by sending raise hand emoticon first, and I will let you to say or deliver your question to our speaker. Or maybe you can write your question in the chat box and I will read it for you to our speaker to maybe give some answer or response. Yeah. And the last, for the convenience during the session, please keep your microphone mute when presentation is going on. Okay, so now let's dive into our topics. But maybe uh, before we begin, before we before the presentation begins, allow me to brief introduce our speaker for today. Maybe the operator, can you present the CV of Dr. Jaspreet first? Okay. Yeah, our speaker today, Associate Professor Dr. Jaspreet Kaur Dhanjal, is an Associate Professor of Computational Biology at Department of Computational Biology, Indraprastha Institute of Information Technology, Delhi, India. Her expertise are biotechnology, bioinformatics, and computational biology. And Dr. Jaspreet already published 53 scientific articles about her expertise. And her recent research is about TCR ESM employing protein language embeddings to predict TCR peptide MHC binding. So for today, Professor Dr. Jaspreet will discuss with us about innovation and development of bioinformatics and the role in education. The role in education. So now allow me to welcome Associate Professor Dr. Jaspreet Kaur Dhanjal before the session begins. Good morning, Dr. Jaspreet. How are you doing today?
I'm doing great. Good morning to all.
Okay, thank you so much for joining us in here today, Professor. And we're really excited to learn your lecture today. Are you ready to begin the presentation, Doctor?
Yes, sure. So shall I share my
Okay.
screen?
Yeah, you can share your presentation maybe
So,
first. And
yes.
for
So is
operator,
it
maybe you can spotlight the Dr. Jaspreet.
Is my screen visible?
Yes, very good.
And my audio is clear?
Yes, very good, Doctor.
Okay.
Okay, without further ado, please welcome Dr. Jaspreet. The time is yours.
Thank you. So, a very good morning to everyone. And thanks for the kind words of introduction. So with this, I would like to begin today's session, and we will be talking about, in general, what bioinformatics is and what all developments have been made in the field of bioinformatics. I'll give you what type of different analysis can be done using bioinformatics and some of the examples from the research which is going around or in my lab to discuss the same, right? So to begin with, who am I? I come from the capital of India, that is Delhi. And my institute where I work as an assistant professor, it is known as Indraprasth Institute of Information Technology. It is a beautiful campus. If you happen to visit Delhi, please do come and visit us at IIIT Delhi. And at IIIT Delhi, I head a group called the Translational Biology Group. And in terms of research, I am mainly a health informatics person who works as at the interface of computational and molecular biology. And if I talk about the work that we generally do in my group, I would categorize into two different, broad themes. The first one is precision and personalized medicine, where we try to look into the disease pathologies in context of individual patients. So we try to understand diseases better, that how a disease affects one person more in comparison to the other, or the therapies that we try and give, why they respond in one of the individuals with more efficacy, effectively and not in the others. And also, can we design new therapies to for the patients, and decide, okay, this subpopulation of the entire disease cohort will be benefiting from this therapy more. So these kind of decisions are something that we try to work for. And the other domain of my research is drug design and discovery, where I'm mainly focusing on proving the potential of the traditional medicinal systems using modern science. So as per WHO, 80% of the world population still relies on the traditional medicinal systems for their healthcare needs. And like even I was in Indonesia a few weeks back, and I got to know that Indonesia also has a rich tradition of traditional medicinal system. If I'm not wrong, it's called Jamu, right? And similarly, India also has a very rich traditional medicinal system called Ayurveda. And it has been practiced since, many years. And we know that these traditional medicinal systems have a lot of potential, but it is still not accepted globally because of multiple reasons. One being that we do not know what exactly happens inside the body upon consumption of these traditional formulations. So if we can use modern science and prove the mechanism of action of these traditional formulations, then in that case, we'll be in a better position to get it accepted at a global level. So these are the two major themes. But I will be showing you a few parts from my research as we move forward, but the topic is not mainly to focus on the research that we do in our lab today. So to begin with, what is bioinformatics, right? So as you can see over here, it is a interdisciplinary science or study, and it uses computation to extract knowledge from biological data. So this is the traditional definition of bioinformatics. We are using computation to extract information or knowledge from biological data. And during this entire process, there are multiple things that are involved. As you can see, there are multiple keywords listed over here: collection of data, storage of data, right? Then efficient retrieval of the data. Then once we have data, can we use that data or manipulate that data for extracting knowledgeable information? Then modeling of this data for further analysis, visualization, and prediction with the help of development of algorithms and softwares. So it almost covers everything. So whatever information is available in the biological data which is available, we want to collect it, right? Collect it in a manner that it can be easily retrieved when needed, right? So we know what is already known and where are the gaps, so that we can choose what we want to work with. And the storage itself, we know the biological data is very, very huge, right? So the storage has to be very efficient. And then again, as I said, analyzing this data, because this is the gold mine. Once you collect all the data together from the biological systems, it is kind of a gold mine which can give a lot of information. So how do we use the available data for analysis and to infer knowledge out of it, and then use it for making certain decisions? And for this analysis, we require a lot of algorithms and softwares that can help us look at this big data. Right, so we will see how big is biological data in a few coming slides. But then again, manual search through this database is somewhat near to impossible. So we need algorithms that can assist us in using this data and analyzing it further, right? But when we are talking about bioinformatics, I said biological data. So what data are we really dealing with? So there are multiple, different types of data that we can talk about. The very first thing is the basics of every living system on this, earth, that is the DNA, right? So when we talk about DNA, we know there are so many different living forms available across around us. So what is the hereditary information that they are carrying, right? The sequence of their genome. Then what makes one living organism different from the other? What are the differences between the genomes, or what are the variations between the genome? Then what are the genes which are responsible for the the genotypic and phenotypic behavior of a living organism? Then not only among different organisms, within one type of organisms, what are the differences in the genomes of different individuals, right? Then when we talk about the presence of different types of genes, then the expression also comes into picture. So when we are talking about the expression of the genes, right? And then we know that the genes, when they are expressed or when they are transcribed, they result in the mRNA or the other non-coding RNA, and then they further get translated into proteins, and proteins are the actual players responsible for the behavior of the cell. And not only proteins, but small metabolites, which are a part of a lot of chemical reactions or cascade of chemical reactions, which are being carried out in the cell almost every instance of time. So the expression data for the genes, proteins, and metabolites, that tells you a lot about the cell, right? Then within that also, say I'm talking only about the proteins. So we know there are like plenty of proteins in the body. So how are they different? What makes them do different jobs in the cell? So analysis of the sequences, the structures, like grouping them, characterizing. We know that we have different families of proteins. We know that the specific protein function of a protein depends upon the motifs it carries or the structural folds that it can take, right? So all this analysis also comes under bioinformatics. Then, as I said, the molecular structure is important, right? So when we specifically talk about proteins, we say that the tertiary structure of protein is very, very important when we are talking about the function of the protein, right? So talking about the structure of the proteins or the small molecules and how this is helping it perform its function, right? Then the chemical biology, which involves all these different biomolecules which I've been talking about. Then talking about everything together as a system and how it is being projected, right? So these are the different levels of data that we are talking about when we talk about bioinformatics, and all this comes under the biological data, right? So when we are talking about bioinformatics, it's not just one skill. It's like a collection of multiple skills that you have and you become a bioinformatician, right? And definitely biological knowledge is the basis for all, right? Now, as I have been talking about a lot of data, you talk about any data, and specifically the biomolecules, we can have two different levels of information. We can have sequence-based information and we can have structure-based information, right? So when we are talking about the proteins, right? So we know there is primary structure of the protein, which only tells you about the order in which the different amino acids are appearing and what is the total length of that particular sequence, right? So when we talk about sequence analysis, it can help us infer a lot of different information. For example, if I'm talking about any protein, I can analyze the physicochemical properties of that particular protein. So what is the amino acid composition? What is the total charge on that protein? The protein, what are the hydrophobic regions of the protein, right? So depending upon the composition of different types of amino acids, a protein can take a different fold, right? Or it could be present in a different environment in a cell. Some proteins could be in the membrane of the cell. Some are there in the cytoplasm of the cell, right? So like this, the location of existence of a protein within a cell also depends on the amino acid composition that it has, right? Then, as I already mentioned, the conserved regions within the protein, the motifs, the profiles, or what are the cleavage sites within a protein when we are talking about its degradation, or some proteins exist in two different forms as precursor protein and then they get matured upon some kind of cleavage activity. So all this analysis can be done with the help of the sequences of the protein. And then again, the phylogenetic tree construction. If I'm looking at the origin of that protein, like the other similar proteins, or the way this protein has evolved in the current state. So all this can be done with the help of the sequence data. Then when we come to the structural data, as I already said, the proteins cannot function till they adopt a three-dimensional structure, the 3D structure or the tertiary structure that we talk about. And for some proteins, even the tertiary structure, like once it is folded, there are multiple subunits that come together and then only it can perform its function, right? So, and not only a like multiple subunits of the same protein, but it could be different proteins interacting together, making complexes and then performing their function. So how there is interaction between or within this protein, or how this protein is interacting with the other different proteins and how it is mediating the biological functions. So this is what we can do with the help of the structure. Then we know that if I'm talking about the normal function of a protein, we see that there are a lot of pathological conditions which arise because of mutation in these proteins. So this effect of mutation on the protein structure and ultimately its function, it also depends upon how these mutations are distorting the structure of the protein, right? And then when we talk about a protein's involvement in some disease pathology and you would want to target that protein, then we want to inhibit the function of that protein. For that also, we want to know the structure of the protein. What are the sites in the proteins where the small molecules are coming and binding? And we would want to target the same site, which is the site where the natural substrate binds. And therefore, we would want to look into the active site of the protein and then design the drugs accordingly, right? So when we are talking about the different biological data, we deal with sequence data as well as structural data. And I'll show you some of the case studies that will emphasize on the same thing again, right? So before we move to the actual analysis part, as I said, the very first keyword that you saw in the definition of bioinformatics was the data, right? The collection of data, the storage of data, and retrieval of data. So these are like some of the examples where the collection of data has become very, very important and useful, that you cannot avoid visiting these collections of data for any type of analysis that you're performing, right? So one of the biggest resources that we have today is the NCBI, right? So we know this NCBI, it actually hosts a lot of different databases, as you can see on the right-hand side, right? So a lot of biological data gets generated every day, right? So many labs are working, so much of data gets generated across the globe. So all this data has to be put under one umbrella or one platform so that it is available whenever it is required by the other people who are working on similar problems, right? So you talk about any gene that you would want to find information about, any protein you would want to find information about, what all genomes have already been sequenced, what all variations are already known for a given protein, right? What all diseases are known, what all genes and proteins responsible for a particular disease condition are known. So everything can be found under one platform, which is being hosted by this NCBI, right? So this is one of the biggest resource. Not only that, the literature which is published, PubMed, right? So for any research article that we would want, the first thing is that we go and access it through PubMed. So all this information is available under NCBI, right? Then another popular resource that we talk about is UniProt, right? So it's like a universal repository for the proteins which are already known, right? So if we look into this UniProt, you will see that it also has different sections different sections. For example, one of the sub databases that has all the like all the proteins which are out there in the literature. But then there is a small section of the database that contains only reviewed proteins, right? Which is very, very important. For example, over here, SwissProt. So it has more than 500,000 entries till date. So all these information, all the information under this section of the database, it is all manually curated and reviewed, right? So this is like a gold standard data about the proteins that you talk about. So I search any protein which is already known. Say I have just put one example. I'm talking about P53, which is one of the tumor suppressor proteins and one of the important targets for multiple cancers, right? So if I just look into this entry, again, this is a manually reviewed entry, as you can see over here. This is the sign for the SwissProt database, right? And what more information can you get about it? So what is the protein, what it is called, what gene it encodes for, like, as I said, what is the status? It is like a SwissProt entry. That means it is reviewed. Because I'm talking about proteins, what is the length of this protein, that what are the evidences available for this protein's expression, right? Not only that, the multiple names and synonyms it might have, in what locations in cells it is expressed, what are the diseases which it is associated with. And then, as I said, mutations, they play a very important role in making a protein, responsible for the onset or progression of any disease, right? For example, we know P53 is a tumor suppressor protein, and one of the proteins which is highly mutated in cancers, right? Because of mutation in this tumor suppressor protein, the cell cannot any longer fight against the transformations that it gets, right? So all these variants, the information about all the evidences which are already there in the literature, it compiles all together. What are the post-translational modifications this protein undergo? The expression of this protein in different tissues or different conditions which are already known. What are the different proteins it can interact with? So, and what are the structures known for this protein? What are the families and domains, the sequence or different isoforms it might have, and similar proteins? So this is like one universal platform where you can get information about proteins, everything, almost everything, under one page, right? So this gives the sense that why collection and retrieval of data is very, very important. So anyone working on P53 can simply access this collection and can get information, whatever they need for their work, right? So this was these two were like the sequence databases. And when we talk about the structural and data, right? So I said information can be in the form of sequences or structures. Both are important. So this again, PDB or the protein data bank, this is one of the biggest repositories for the structural data. All the structures that have been solved for proteins or DNA structures or RNA structures, their complexes with small molecules, protein complexes, everything that has been solved till date, it is available under protein data bank. As you can see over here, more than 200,000 structures are already available. And not only that, as you know that computational structure prediction or computational protein structure models, it has gained a lot of attention in the recent decades, right? So people try and predict the structure of the protein if it has been not experimentally solved. And there are very good methods available today which can make structures, and it has been proved or it has been tested that the structure predicted is very close to that of the experimental structure, right? So all the structures which have been computationally predicted now are also stored under PDB. Right, so you access any page, you will see that you also get structure which is predicted by AlphaFold, one of the AI-based method for the prediction of these structures. It is based on large language models, right? So again, PDB, very, very important resource. And this is like one entry from PDB that I'm showing over here. So again, this is a P53 itself that I was talking about in UniProt. So this PDB, it gives you all information. What was the method used for finding this structure? What is the resolution of this structure, right? And what all different structures are present, like in terms of biomolecular structures, what all entries are there. For example, this structure, it has two chains, both are P53 structures. And then what is the sequence length? If at all there are certain mutations in the natural sequence of the protein, like sometimes the protein is difficult to be crystallized, and some mutations are introduced to make it stable. So similar kind of things, or it can be a mutated structure itself which has been solved. So all this information, if at all it is complex with a small molecule, you will also have entry for small molecules or heteroatoms. So if, say, I am talking about a structure in presence of a drug molecule, right? So a drug interacting with the protein. So I do get co-complex structures in PDB. So all this kind of information is available under PDB. And not only one, if you just search P53, you will get multiple structures within PDB for just P53, solved by different groups, different variants of P53, different lengths of the proteins crystallized. So if you are actually choosing a structure to work with, you really have to make a decision which structure you want to go with, right? So these are some of the gold standard databases which almost every biologist uses for their work, right? But not only these, like these are just three that I mentioned. There are like thousands and thousands of databases available online. And depending upon what work you are doing, you might also make some custom databases. And as I just mentioned, this time's Nobel Prize, it has gone for the computational protein design and protein structure prediction, right? Computational work. So that's why I said that this is important. And therefore, PDB also has one section for the computationally predicted protein structures, right? So you can actually understand the importance of computational or studies or bioinformatics in today's biological world, right? Okay. So as I said, not only the standard databases which contain information which is used by almost everyone, there can be some custom made databases also. For example, this is like a small part I am showing from my own work. So as I said, I'm interested in science related to traditional medicinal systems, right? Or medicinal plants. So one of my aims is to find out all the medicinal plants, what are the small molecule present in these medicinal plants which are responsible for the therapeutic potential, right? So for example, a lot of research is going on these days. For example, if you look at this paper from 2022, this is one of the reports from one of the paper published in Scientific Reports. They have worked on this plant called fenugreek, and they have identified what are the different bioactive components present in this plant and what are the therapeutic activities associated with those, right? So for example, these flavonoids, these are very good source of antioxidants. Again, they have anti-inflammatory property. These can be neuroprotective. Similarly, say if I talk about these saponins, again, these are also anti-inflammatory. They also have anti-cancer property. So a lot of work is going around for medicinal plants, right? So people are understanding the importance of proving the therapeutic potential of these plants. But when I talk about the medicinal plants around, right? So this data is from 2006. I could not find a more recent report which compiles this type of data. But if you look at, this is very old data, but still look at the numbers. So these are the plant species known from these different countries. And this is the count of medicinal plants which have been characterized out of all the plant species, right? So say 21% of the plants found in Bulgaria, they have medicinal properties. And if I look at the total number of medicinal plants known during that time, or I would say the categorized during that time, it was 72,000. So much of work is going around about all the plants. Say if, even 1,000 of plants have been characterized in the form that I'm showing you on the left-hand side, how much information is available outside, right? Like in the world. And as a person who's working with medicinal plants, I would want all this information to be available at one place so that whatever I want to know, okay, this plant is already characterized. I already know about the therapeutic properties of these plants, right? So what am I looking at? Something like this. I've been working with ashwagandha from last 4 to 5 years. And I'm also focused on the anti-cancer activity of ashwagandha. So this is an image from a review article. So it actually compiles everything that is known about the anti-cancer activity of ashwagandha, right? So what all are the different molecular players which go up or down, and they mediate the different, or what hallmarks of the cancer do they really target, and how they are mediating that activity, right? So this is what I am expecting. But this information is, all this information together is not available in one place. We do have a lot of databases. For example, I have listed some of the databases over here, which have data related to medicinal plants, right? And I have listed some here also, and how many number of phytochemicals they have at present, right? But then again, when we started looking into the literature, we realized there is much more information available out there. And with time, the research on medicinal plants has actually, started, like more and more people are working on medicinal plants. So every day, a lot of information gets generated and gets published in the open repositories. So our aim is to bring all the information which is available till date under one platform. So we are actually in process of making this database, which can contain, secondary metabolites from different living sources, and we want to compile them all together, add what information is already available. For example, what is the bioactivity associated with those secondary metabolites? What are the molecular targets already known? So everything can be put under one platform. So whosoever is working with natural products can access this kind of repository and can benefit from that, right? So this kind of gives you the importance of the data collection, data storage, and easy retrieval, right? And say if someone is trying to find out a compound for antioxidant properties, they can actually use this available repository, look into all the secondary metabolites available from different natural sources, and then they can do a drug repurposing, right? So everything becomes available if the information is available under one resource, or under one umbrella, right? So from here, we again move little forward. So this was more about data collection, data storage, data retrieval. Now, once the data is available, what can be done, right? So a lot can be done once the information is available, right? So I just picked up this small example because we all know about SARS-CoV-2, right? We all have been affected by the SARS-CoV-2 directly or indirectly very recently, right? So how this actually started, the research around SARS-CoV-2, how was it identified, what kind of research was carried out, and how the available data actually helped in that analysis, right? So this is like one of the reports, very early reports during SARS-CoV-2, right, where they identified SARS-CoV-2, right? As you can see over here, they extracted RNA from the lung fluid of the patients, and they did the Illumina sequencing and nanopore sequencing, right, to characterize the virus genome, that what was the virus which was infecting these patients. And then all these reads, whatever they extracted, the RNA that they extracted, they sequenced it, and then they tried to identify what virus it is, right? So for this, I will directly, go to the next slide. So as you can see over here, they identified that the SARS-CoV-2 that they were looking at, it had 80% identity with a bad SARS-like CoV-2, right? As you can see over here, as if you remember, then the slide that I showed you for the protein sequences, I told you that we can use sequences for phylogenetic analysis. So this is an example of the same. They used the sequences, they characterized they got the sequence for the viral genome, and then they used that viral genome, which was approximately 30 kb in length, and they used that sequence and they did a phylogenetic analysis. And as you can see over here, so these were the sequences that they generated from different patients who were infected by SARS-CoV-2. And as you can see, the closest among these were the SARS-CoV from from bat, right? The bat SARS-CoV. So from here only they identified that 85% identity was there in the genome of the SARS-CoV that lives in bats or affect bats, and this new SARS-CoV, right? So from this only this sequence analysis, they, as I said, they did sequencing, they assembled the entire genome of the virus, and they identified, okay, this virus is new, but it does have similarity with the viruses which have already which were already existing or which were already characterized, right? So this gave us a platform to start with, right? Not only that, when the different variants came, right? So we said, okay, this variant was had more like could transmit more, or this variant had severe effect on the patients, right? So a different type of analysis, again based on the sequencing, was done. So how was one virus isolated from one patient different from that of the other, right? So a lot of sequence analysis, as you can see over here. So it is telling you about the different samples from which the viral genome was extracted, and then what are the positions that were varying among us, right? So what are the synonymous and non-synonymous mutations that they observed? So this is a very small representative data, but analysis at a large scale actually told them about the differences that they were observing among different variants of SARS-CoV-2 which were out there, right? And they could also find out the reason why one of the variants is causing more severe symptoms in the patients in comparison to the other, right? Or why was the transmission more for one of the variants than in comparison to the other? So this all could happen because we could generate the sequence information of the genome of SARS-CoV, right? Not only that, once the genome was characterized, we knew that it is similar to bat viruses. Then they also could, because there was 85% identity, they could also infer which regions of the genome of SARS-CoV-2 were responsible for making what type of proteins, right? So once we knew what this virus is all about, we had the options to also look for drugs against this SARS-CoV-2, right? So this again is one very small example where sequence analysis was the primary key, right? So if you remember, there were two there were multiple proteins which were characterized. So if I say like main protease was one of the, or MPRO was one of the proteins which they found was very important for the survival of the virus, but was absent from the humans. So this became one of the perfect targets for designing drugs, right? And as you can see over here, there are multiple viruses which showed some kind of identity or similarity with SARS-CoV-2. So this is just one sequence alignment for all the main of the main proteases from these different SARS-CoV-2 and its alike viruses, right? So as you can see over here, some regions have been marked, right? So there were already known drugs against main proteases for different viruses, right? But as you can see over here, the structural similarity is quite high, right? Also, the drug interacts with some of the important residues in the active site of the protein, right? And these marked are the amino acids which were responsible for interaction with the drug. As you can see over here, these regions were highly conserved among, like they're almost identical among all the different main proteases from all the different sources that we were looking at, right? So that means it says that the active site of this main protease, it is being conserved among these different viruses that we were looking at. So if we have a drug against main protease of one virus, we can repurpose it for the main protease of SARS-CoV-2 also, right? So we did had a few molecules being tested against main proteases of different viruses. So it could be checked against SARS-CoV-2 as well, right? Then the other example, I think everyone can relate to it, this RDRP. So this is also one of the proteins which they found to be conserved among different viruses which they found similar to SARS-CoV-2, right? This RDRP. Then we all know about this drug called remdesivir, right? So this drug was already known to work against RDRP from similar viruses, right? So we knew that because SARS-CoV-2 also has this protein RDRP, and it is similar, or the sequence is very similar to that of the same protein extracted from other viruses. So we had some confidence that remdesivir can also work against the RDRP of SARS-CoV-2, right? Again, a good example of drug repurposing based on the sequence analysis that we had done. And not only sequence, like once you know the sequence, you can always go to the structure and look into the structural similarities also. If there is no difference in the active site where it actually goes and bind, in that case, we can use the drugs which have been, identified for some other task before, right? So this is again an example of drug repurposing based on the sequence and structural analysis that we did for the SARS-CoV-2, right? Now, when we talk about bioinformatics, again, when I'm talking about all this information, whatever I just talked about, it was because we already had some information available and we made use of it, we did some analysis and we came to some conclusions. So similarly, we can also use bioinformatics for computer-aided drug designing. I just gave you some examples. I'm elaborating a little bit more on that, right? So when I'm talking about drug designing, either you have some clues. For example, we had some clue, okay, RDRP is being synthesized in SARS-CoV-2. We already have a drug known against RDRP of similar viruses. So we can use this remdesivir against RDRP for SARS-CoV-2 also. But say we do not know what drugs, or there are no similar, proteins or drugs available. So in that case, what do we do? But we know that this protein is responsible for causing this specific disease or disorder, right? So can we design some new drugs against it? Yes, we can again use bioinformatics and do the same. So we can have two different strategies. We can have structure-based drug design, right? Say I have discovered a new disease, right? A new disease has come up and I identified the causative, the causative agent, say some pathogen for that. And I've also identified what type of proteins that pathogen make and which are absent from the human cell, right? So that means those are like good targets. If I block the function of those proteins, that pathogen might not be able to survive, right? So what I have is the structure or the information about the proteins that this pathogen is having. Now, again, depends. Do I have the structure available for this protein from pathogen? If yes, I can use the structure of the protein and look for the drugs that can inhibit, right? So that becomes structure-based drug design. You already know what molecule you want to target and you are designing small molecules for that, right? So with known protein structure, as I said, if I already know the structure, I can simply look into the small molecules which are available, right? So I listed 3 important databases. When I'm talking about small molecules, we again have some very good databases available for that. One of the repository which has a lot number of small molecules is called PubChem. Then we have Zinc repository. So if you look into any paper which talks about virtual screening, drug designing, they will talk about these databases, right? Then you have drug bank. So these are some of the standard databases which people use. But not only these, we have many other for small molecules. So we can actually, what we can do is we can take these small molecules and we can see which of these small molecules have the potential to interact at the active site of the protein. So for example, over here, this green is one of the small molecules. So we can actually use the process of virtual screening. So we have the protein, the structure, we have the structure of all the ligands that through which we are trying to identify that candidate molecule. We dock all of these small molecules one by one, and we see which of these fit well and has a good interaction energy. And that could be one of your leads to begin with, right? And then if we do not have known ligands, say we do not have these ligands available, and we want to design altogether a new ligand, then we can actually look into the active site of the protein. We see, okay, here I have one OH group, right? So there are some hydrophobic amino acids over here, right? And then accordingly, we identify some functional groups in the cavity of the protein, and we try and fit certain fragments which can interact with these groups, and then we try to stitch them together into a molecule, right? So there are again algorithms to do the same. We identify the functional composition of the active site. We identify fragments, we stitch them together, and we see if the molecule is synthesizable, right? And if yes, then we can synthesize and test whether these molecules can inhibit the functional activity of the protein or. right? But then there can be other cases. Say the structure of the molecular target is not known, right? For example, there are a lot of studies in which, say, I'm talking about one bacterial cell. I have number of ligands which I know that work against this bacteria, but I do not know which particular protein of the bacteria it is actually targeting, right? So I just know that these ligands or small molecules, they're effective against my protein, my organism, but through which protein, I have no clue. In those kind of cases, like this is just one example, there can be multiple examples. What we used is ligand based drug design. So as I said, I already have a set of ligands. Say I already know 100 small molecules, and if I expose my bacteria to all these 100 molecules, it just dies, right? So that means these all ligands are effective against the bacteria. So I can actually use all these ligands, I can learn from these ligands, and I can design new drug candidates, right? So we have, again, different algorithms for to do that. We can do similarity based search, exactly similar looking molecules. We can do pharmacophore based screening, I'll come to that. We can, once we have some molecules, we can change the structures at a few positions, and when we can see whether we can increase or decrease the activity. So we can have models which find out relation between the structure and activity of small molecules. It is known as quantitative structure activity relationships, QSAR models, right? But yes, if we do not have any information about the the target protein or the small molecules, if none of the information is available, it's difficult to get to start with computer aided drug designing. But overall, computer aided drug designing has accelerated the process of obtaining the lead molecules, right? So we do some kind of, we collect some kind of knowledge, we build on that knowledge, come up with some small molecules, okay, and then we say, okay, these molecules may interact with your protein or may inhibit the, say, bacterial cell, as I gave an example, and then in experiments, we can test the same, right? So again, bioinformatics plays a very important role. You are using already available data. You are using certain algorithms to predict interaction between proteins and small molecules, or compare already known small molecules with a list of new molecules, right? And then you are coming up with lead molecules. I just give you an example again from SARS-CoV-2, right? So once all this analysis was done related to SARS-CoV-2, we got to know that there are two potential targets for designing drugs, which almost everyone was talking about. It was this TMPRSS2, and there was another protein called ACE2, right? So almost everyone was trying and designing drugs against these. So these were like two of the receptors which are expressed on human cells and were responsible for the entry of the virus into the human cell, right? So there was an hypothesis that if we block these receptors, then the virus won't be able to enter into the cells, right? So a lot of people were working, and we as researchers also wanted to look into it. And again, because we work with like small molecules from natural resources, and there are already known properties of ashwagandha, cape, which are like natural bioactive compounds, that they do have antiviral activity. Not against SARS-CoV-2, because this was identified for the first time, but I knew that ashwagandha has antiviral activity. It has been used against viruses before, right? So we thought, why not test these natural molecules for SARS-CoV-2 as well, right? So we did a screening against TMPRSS2 as well as ACE2. So I'm giving you example of only TMPRSS2 over here, right? So again, drug repurposing is, this is what is known as drug repurposing. So we identified the target, as I said, ACE2 and TMPRSS2, they were known as the two targets. Then we looked into the literature to find out whether there are already available drugs which can target these two receptors, right? So we found one structure in PDB, one structure, this called chemostat, which was known to inhibit TMPRSS2, right? So we had the structure of TMPRSS2, we docked chemostat to that structure, and we saw how this chemostat, which is already known inhibitor of TMPRSS2, it interacts with the protein, right? And once we knew how this chemostat, already known inhibitor, is interacting with the protein, we actually docked multiple small molecules from natural resources to see if they can bind to TMPRS2 in exactly the same way as chemostat does, right? So as you can see over here, I also talked about ashwagandha. So these are two bioactive molecules present in ashwagandha, right? Vidafirin A and Vidinone. So this study is only looking into the bioactive compounds from ashwagandha. So as you can see over here, this red one, chemostat, it was interacting in this pocket. And these two molecules, they were also interacting in the same pocket of TMPRSS2, right? And there was a report in literature where they had discussed the mechanism of interaction of this chemostat with TMPRSS2. They said once it interacts, they see some conformational changes in the protein. So they saw the formation of these alpha helices, right? So similarly, when we docked over Vidafirin A and Vidinone and then simulated the structures, so we also observed these changes in the structure of the protein, right? So we had little confidence that yes, Vidafirin A and Vidinone, it can behave in a manner similar to chemostat against TMPRS, right? Now, during this time, I had mentioned about simulating the structure, right? So this is again an important approach in bioinformatics, molecular dynamic simulation. It has a lot of different applications, but the idea is the same that I'll tell you now. So when we dock the small molecules into protein, so I just take one protein, one structure from PDB, or I model my own protein structure. Then I identify the active site and I dock my molecule into that active site, right? And this is all static. My protein is static, my ligand is static. I'm just putting my ligand in. And there are advancements into algorithms which make some part of the protein flexible. The ligand can be made flexible, and I can see which is the best conformation that it fits in. However, when we talk about these proteins in our body, they are always in dynamic motion, right? They keep, because we have a lot of water present in our cells, or the environment is aqueous, right? So it keeps on moving, or it is always under dynamic changes. So to replicate the same, or to some extent, we do not replicate it entirely, but to some extent, to mimic the same conditions, what we generally do. So before we move forward, this is just another representation of the same. So which is showing that this chemostat, it is interacting with multiple residues, as you can see over here. Serine 441, right? This aspartate 345, right? This histidine 296, right? So these are like some of the residues which you can see almost everywhere, right? So this one. So as you can see over here, so these residues, they were present in all the three structures. So that means we had this confidence that it is interacting in a manner similar, that like these two molecules, Vidafirin A and Vidinone, they're interacting in a manner similar to chemostat, right? But then, as I said, these were static structures. So what in molecular dynamic simulations we do? So this is, as I said, only one of the applications that I'm discussing. So to mimic how actually these molecules behave in cells, we make a box, we fill it with water, and we put our protein into that box, right? And then we use, again, algorithms to assess how this protein will act inside a aqueous environment, right? So this is what we do. Say this was my TMPRS, though this is not TMPRSS2, but assume that this is TMPRSS2 and this is docked with Vidafirin A, right? So I have a small molecule attached over here, and this is my Vidafirin A. So I will put it into this water box, let it be there for some time, right? So we run molecular dynamic simulations for very small time frames, a few nanoseconds, a few microseconds. And then we say if it stays in the same pocket or not, right? Because of the dynamic motion of the protein, this might not be strong enough interaction that Vidafirin A can stay here. But if it stays throughout the simulation period, we say, okay, the interaction is strong enough, right? So we did this kind of analysis, and then we looked into the different residues, right? I just showed you three. Histidine 441, serine 441, histidine 296, and aspartate 345. Right? So we do this kind of analysis and we see which are the important residues, right? So which of the important residues are present and how your small molecule is interacting, what type of interactions it is making, right? So hydrogen bonds, hydrophobic interactions, ionic bonds, water bridges. they all help a small molecule to interact in the active site. So how are these interactions keeping that ligand is something that we have done some analysis. So again, very representative data. We do a lot of different analysis to establish that yes, our small molecules or these small ligands, they do interact with the protein and the interaction is stable, right? So once we get certain leads, we can actually move on to the next step. We can do in vitro analysis or in vivo analysis. This is just one example. So not only Vidofrin A and Vidinone, we actually tested a lot of different molecules, right? So this is Vidofrin A that I was talking about. This is WIA that I showed you. And then we had Vidinone. So this is WIN that I showed you. So we tested multiple molecules. We did docking, we did simulations, and we came up with some. So this list is small. So these are the ones which showed interaction the TMPRSS2 in the docking study, right? And they stayed in the same pocket in the molecular dynamic simulations also. So we thought, okay, let's test them in vitro. Now that they're showing interaction, we can test them in vitro. So we did some in vitro analysis using cell lines that express TMPRSS2. And then we saw that it leads to the decrease in the expression, right? As you can see over here. And also the, it is inhibiting the protein activity also. And as well as it is downregulating the expression of TMPRSS2 at the transcription level also, right? So this is again very representative data, very small data. We tested it in multiple cell lines which express TMPRSS2, and we saw how these molecules, they were interacting. Then again, so we got some leads from our in silico analysis. We took these leads to in vitro analysis. And then again, we shortlisted some molecules and we collaborated with a lab who were actually working with SARS-CoV-2, right? So they took some cell lines again, Viro E6, and the molecules that we had predicted and tested in vitro, we gave them those molecules, and they saw if these molecules can actually inhibit the entry of virus into the cells. As I said, TMPRSS2 and ACE2 are two receptors which are expressed on human cells, and they are involved in the interaction of virus and the entry of the virus into the cells, right? So as you can see over here, this is percentage inhibition. So after 48 hours, this is the percentage inhibition. If you remember, I talked about remdesivir, right? So the protein target is different, definitely. But again, this is also one of the molecules which they say can inhibit the SARS-CoV-2. So remdesivir, it showed 99% inhibition. And for the molecules that we tested, we saw a range of inhibition, right? So some of the molecules were really effective. For example, this vidinolide B, 82% inhibition could be seen. Then Vidinone, it showed 66% inhibition, right? So similar. So these are like some of the molecules which actually showed some potential. So not only that, these are like pure compounds. We also saw if we can directly use extracts from ashwagandha, right? So this analysis also we have done, and we have tested in Viro E6 cells using SARS-CoV-2. So we actually suggested that I'm not putting it as a therapy for SARS-CoV-2, but it can be used for the management or prevention of infection, right? So not only ashwagandha, so these are all bioactive compounds from ashwagandha. We tested small molecules from other natural sources also. So there were a few molecules that it is all published and available, that there are some natural molecules which inhibit the entry of SARS-CoV-2 into the human cells. And if administered as a, like we are suggesting that if we can take it as these extracts, it can help us in preventing the infection. Right? So again, this is one of the examples where we see that the in silico analysis or the bioinformatics analysis was the background or was the first step which helped us to build onto these. Right? So not only that, as I said, I'm working with the natural bioactive compounds from almost 5 to 6 years now. I think I have more than that. I started working on these during my PhD. So during all these times, I have looked into bioactive compounds from ashwagandha, honeybee propolis, marine carotenoids. Now we are working on blackberry lily. So these are like different natural sources. We have extracted small molecules from them. For example, as I just showed you, Vidofrin and Vidinone are two of the bioactive compounds we very frequently work with. Then the scape from honeybee propolis, fucoxanthin from marine carotenoids, and pectoragenin from black lily. So this is a very limited list. We are working on a lot other molecules also, and using the approaches that I showed you, right? Virtual screening, molecular docking, molecular dynamic simulations, and also our gene expression profiles, we have been able to identify that these are some of the molecular targets or proteins through which they mediate the activity that I'm talking about. Say anti-cancer activity, anti-metastatic activity, anti-COVID I just showed you, then neuroprotective activity. So we are working with this compound, pectoragenin, and there is another compound, alpha lipoic acid, which has neuroprotective activity, right? So again, we start with some clues from literature, we build using computational approaches, and once we have some good leads or information, we try and test them in vitro. And if it works in vitro, we collaborate and we do some in vivo experiments, and then we present those molecules as potential therapeutic molecules, right? So this is one example. What I showed you before this it was all structure-based screening, right? We already know the structure. As I mentioned, in computer-aided drug design, we can have structure-based drug design or we can have ligand-based drug design. So what I showed you till now, they were examples of structure-based drug design. So this is another example where we do ligand-based virtual screening, right? So again, this is one of the works that are presently being done in my lab. So we are working on a disease called Wilson's disease. So in this, there is excess accumulation of copper in the cells. So our body has one transporter which senses the presence or absence of copper in the cells. And like there are two different transporters. So they help in absorbing the copper as well as excreting out the excess copper if it is present in the cells, right? So in Wilson's disease, the transporter which senses the excess copper and removes it from the cells, it is mutated, right? So again, sequence analysis has been done, a lot of sequence analysis, and we know that there are more than 500 protein variants of that transporter that removes excess copper, right? So these 500 protein mutations are responsible for causing Wilson disease, more than 500, right? Now, the available cure or therapeutic options is that we use some small molecules which chelate with this copper, convert them into a form that can be excreted out of the cells, right? So these are three molecules which have been FDA approved, but we have some more molecules which are under investigation or in clinical trials, right? So we studied all these molecules, and then here these molecules, they are only interacting with copper, right? So I do not have a protein in question. So I'm using ligand-based drug designing over here. So I'm using these molecules, available molecules, FDA approved, under investigation, and in clinical trials. I use all these molecules and I study these molecules, right? And then based on the features that these molecules have, we are trying to predict new molecules, right? So as I said, either we look for molecule which has a structure very similar to this penicillamine, right? Any molecule which has a very similar structure can also behave like this. So this can be one criteria, that on structural similarity, I pick certain molecule, right? But then we have a better way of doing it, which is known as pharmacophore-based virtual screening, right? For example, this is just an example. This is my small molecule that I'm talking about. This is already known ligand that works, right? Now I look into this ligand, and as you can see over here, right? So this is an aromatic ring. I'm saying that my ligand has one aromatic ring, and I represent, okay, here I have one feature called aromatic ring, right? And then this group, this tail of the molecule should have some hydrophobic residues, right? Some hydrophobic molecules. So over here, hydrophobic region has to be there, right? Then as you can see over here, I have one hydrogen bond donor in this area, and I have one hydrogen bond acceptor in this area. So I mark my ligand in terms of these functional regions in the protein, in the small molecule, right? So the same ligand has one aromatic ring over here. It should have a hydrophobic region here. It has one hydrogen bond donor, one hydrogen bond acceptor, right? But I'm not defining which hydrogen bond donor or which hydrogen bond acceptor. So I do the same for all the ligands, right? So I mark these functional groups in the small molecules for all my ligands. Say I have 10 ligands, I do the same for all. I can overlap these and see, okay, all the molecules have this hydrogen bond donor. All the molecules have this hydrophobic group at the end of the tail. And that means these groups are important for the functioning of these ligands, right? And then we can also see how they are spatially arranged. That means my hydrophobic group should be at the tail. My hydrogen bond donor should be here, and aromatic ring should be on the other end. So the spatial arrangement of these groups. So how are they separated by distance, and what are the angles they are separated with? So this kind of information then I have, big libraries I talked about. We have a big database called PubChem, Zinc, Drug Bank. We can download all the molecules. We can have their structures defined in the same pharmacophore-based way. And then we can actually look into the similarity. So any molecule which has spatial arrangement of these kind of groups, that can be one of the potential leads, and then I pull it out. So we have actually used the same kind of method, right? An algorithm which can help us in pharmacophore-based virtual screening. And we have come up with a list of molecules which can have similar kind of activity, right? And now we are actually trying and looking into the interaction of the new molecules that we have identified, that how they interact with copper. And then once we are sure that they interact with copper with good affinity, they can make the structure that these already known molecules they make. In after that, we will be able to shortlist them further, and then take some of the molecules and test them in vitro again. And if they work in vitro, we can always collaborate for the in vivo experiments, right? So this tells us that one of the major area in bioinformatics is computational based drug designing, where we can have structure based drug designing, or we have ligand based drug design. And we have a lot of advanced methods for doing the same, right? The algorithms that help us in doing these virtual screening, docking, molecular dynamic simulations, they're improving day by day, right? The computational advancement is happening, and we have techniques which give us results close to the reality, right? But then again, these are the screening methods. So once you identify some certain leads or candidate molecules, you do have to go for the experimental methods, right? So this is computer-aided drug design. Now we come to another aspect of bioinformatics, right? So far we just talked about simple sequence analysis, simple structural analysis. We talked about computer-aided drug designing. Now we come to the other advancement in the bioinformatics, that is making use of big data, right? So what do I mean by big data, right? So I'm focusing on healthcare, but you can actually imagine that the same thing can be done for plant-based research, for animal-based research, right? So you can extrapolate the potential of these techniques that I'm talking about in the different, living systems. I'm mainly focusing on the healthcare aspect, right? So when I talk about big data, right, as I said, a lot of research is going around across the globe, and every day we are producing lots and lots of data, right? Say I'm talking about the Wilson that I'm working on. I sequence a few patients, right? I have a few patients who have Wilson, and I see that there are different types of variations between different patients. I sequence their genome to look into what are the DNA mutations that I'm talking about. And then instead of doing only the targeted sequencing, that is for that transporter that I was talking about, I actually sequence their entire genome. But I did not use the entire information. I only used the protein, the specific region from the genome that encodes for that transporter, right? I did my work, and the rest of the data I've already submitted. But I actually generated a lot of data. I generated genome profiles from, say, 50 patients. So this data has a lot of other information, right? So this is kind of a big data, the genome from different patients. So this is just my example. As you see that there are so many different sequencing facilities available, and people are sequencing a lot of data. Not only sequencing, right? So as you can see over here, these are different sources from where we get a huge amount of data in healthcare, right? Again, this is data from a report that got published in 2019, right? So as you can see here, how much data do we produce? So we all are familiar with gigabyte, right? We say, okay, this much gigabyte of data. So when we are talking about exabytes, that means I'm talking about 10 to the power 9 gigabyte is equal to 1 exabyte, right? So this much amount of data. So as you can see over here, in 2013, we produced 153 exabytes of healthcare related data. And then they estimated that this volume of data will accelerate to 2,314 exabytes in 2020, right? We are already in 2024. So you can imagine how much data has been collected over these, 10 years, and how much data is available and what can we do with that data. The potential is huge, right? And what all are recorded in these data? First of all, as I said, sequencing. I just gave you a very small example, but I will actually show you there are sequencing projects, massive sequencing projects going across, in different countries across the globe, right? So even if I'm in India, I am sequencing like 1,000 patients. So you can imagine the total genome, the full genome of 1,000 patients, how much information it carries, right? And for what all applications I can use it, right? So not only that, so many imaging, right? So you go to any hospitals, there are continuous X-rays being taken for different types of disorders, and all of this data is being collected, right? So if you go into the literature and you start looking into what type of work people are doing, you will see that people are doing a lot of prediction mod, like they're generating a lot of prediction models using imaging data, X-ray of lungs, then you during COVID, a lot of people made models that can look into the lungs, X-ray of the lungs, and tell, okay, this patient has the SARS-CoV-2 infection or not, right? Then the health records. So if I, as a person, I collect all my medical records from day one till date, right? It is kind of a medical history for me. Whatever has happened to me over these years, everything is being recorded. And there are a lot of hospitals which maintain electronic records of their all patients. So this data itself, again, is a gold mine. You can mine a lot of information that can be used or translated for a lot of health applications, right? Again, medical devices. So we use so many medical devices. Almost everyone wears a smartwatch, which can record their heartbeat every second, right? Their heartbeat, the footsteps they are taking. Some electronic devices, for example, the wearables that we are using, they have some smart sensors also. Not only the heartbeat, they can actually have a lot of other health related parameters being recorded. So again, this data gets recorded onto a cloud. If we want, we can access these records, right? Then smartphones, again, smartphones also has a lot of health trackers and being installed, and people are using it these days. So again, for those people, you actually get personalized health records, and it's very, very important these days, right? Then the payers record, like the health insurance company, right? So many people have health insurance. So every time they fall sick, they claim, the companies are actually collecting data. What happened to the patient? How the patient, fell sick? What are the treatments they are getting? How much was spent on that? What kind of tests were done? All the reports are being collected. Again, this becomes very important data, right? And then pharmaceutical research. A lot of new drugs are being, worked for. And then the trials, some drugs fail, some drugs succeed. So why a drug failed? All those experiments, the related data, everything is being archived, right? And then again, the internet. This is like one of the biggest resource of data that we have, right? So if I get time, I will share very interesting report, but that is from 2000, I think 2006 or 9, done by Google, that shows you how search engine data, like whatever we are searching in Google, that can be used for tracking or predicting the outcome of diseases, right? Or I can just share that paper. If you are interested, you can go through that. So again, the emphasis over here is the big data, right? We have so much data around us that we can use this data for many, many different applications, right? First is diagnosis, or we can, whatever we are at, like once we go to a doctor, we get an X-ray done, they read the X-ray and then they interpret it. So if we already have a lot of X-rays available, the reports that was generated by the doctors, we can actually use them and create an automated, software. You have any X-ray, the software will work. It will tell you, okay, this X-ray means the person has tuberculosis. And why the algorithm predicted it to be tuberculosis, it actually marks the region. Because of this region in this X-ray, I am predicting, like the algorithm is predicting it to be a tuberculosis case, right? Again, drug design and discovery, predictive analysis, as I said, a lot of prediction can be done. We can optimize dose, right? So we have some data from a pharmaceutical company say, this drug, it worked in this patient, not in this patient. In this patient, I had to give this much dose, but in these 10 different patients, I had to give 10 different doses. So why different doses had to be given to different patients to see the effect? All this optimization can be done, right? And personalized medicine is one of the recent trends which have come up because of the availability of big data. We say no two patients are similar. So we can look into all these aspects, all the data available from different patients, and see why something worked and why something did not work. Right? So again, a lot of applications. Let's see just a few so that we understand how this big data can help, right? So as I mentioned, these are some of the initiatives, population scale sequencing initiatives going across the globe, right? So as you can see here, so many different countries, they are working on sequencing the genome, right? So I've also mentioned about the India Genome Project, right? So they are also trying to sequence a lot of human genomes, and there are a lot, like these are like massive. Why I want to emphasize on this? These are like massive projects. As you can see over here, even if I'm talking about India, these many institutions have come together and they have done a partnership so that they can work for the Genome India project. It's not only sequencing. Once you have sequenced, you generate huge amount of data. You have to store it. You don't want this data to be, available without any control, because this data can actually tell a lot about the people from where it is coming, right? Their health issues, if they are prone to certain disease. So this has to be controlled. It has to be in databases which are under control access. And then again, why people in India also are different from each other in terms of their response to certain drugs or susceptibility to a disease. A lot of analysis can be done. I'm just mentioning a few different cases, but you can imagine if you know the genome or you have the genomic information of say 10,000 people, what all amount of work can you do, right? So this is again one example, already published paper from 2018, right? So here I just wanted to emphasize how big data can give you a lot of information and can improve your decision making powers, right? So no need to get into a lot of technicalities, just kind of a story that I want to tell you. So this is a tool, again a bioinformatics tool called Haplosaurus, right, which has been developed and it can have multiple applications. I'll just discuss one. If you go into the paper, you will see they have discussed a lot of case studies, very different from each other, but it shows the potential that big data has, right? So what they have done, like what this tool actually works on, you have sequencing data from individual, right? You're sequencing the genome, right? And then you know that we have reference genome for humans and other living organisms. So they have sequenced the genome from different individuals. They align it with the reference genome, which is already available, and they try to find out what are the variants, right? So if say it's my genome, then if this is the reference I'm aligning my genome with, at this position in the reference genome, I have a different genotype at this particular position, right? So this is a variant that I carry. But for this position, because we have, our genome is diploid, right? So I can have two different alleles. So for this particular position, I have two different variants, like one which is matching to the reference genome, the other one is different, right? So this is like, if you can get information about the haplotypes also, then it is like phased genotyping, right? So they have done similar kind of analysis. They have used sequencing data from different individuals, aligned it to reference genome, and they have seen what all genotypes are available for different locations in the genome, right? So as for example, over here, one of the chromosome from the individual, like at this position, it has only green color. So that means both the chromosome have the similar allele, right? At this particular position, the reference genome, you can see yellow, but here we have yellow and red. So that means one of the chromosome has the yellow allele, the other one has red allele, right? So like this, for every individual, we can actually see what are the different combinations of alleles present, right? Now, this is about using the sequencing data. The next is about Just give me a second. So next is about predicting the proteins. Now we know that not all mutations result in changes in the protein, right? And for most of the drugs, we are targeting proteins. So can we directly use the information available about variants from the DNA sequence and use it for making drugs? No. We have to see how those changes in the DNA are resulting in changes in the protein, right? Now, say the variant mapping was already done. Say these are the two haplotypes that we have observed for an individual. Now, these haplotypes will be converted into in silico proteins. Right? We know that if we know the DNA sequence, we can translate them and predict what kind of proteins they will make, right? And depending upon what kind of variations do I have, what kind of proteins I can make, that can also be predicted. Now, comparing it to the reference genome, right? I say, okay, for this particular haplotype, my reference genome, say I have sequenced, I did this analysis on 1,000 patients. In those 1,000 patients, only 23% had the genome similar to the reference, right? The one that I used over here, this reference genome, and 65% of the patients, they had mutation, right? They had mutation from R to Q. And then 12% people had 2 mutations, right? So that means at population level, different people are producing different types of proteins. So if you have one drug against one protein, it might not show similar effect, as I was discussing before, right? The same gene, but different proteins are being formed if you look at the population level, right? Because of the different variations that can exist there and the combination of different variations. So if I say there are 5 SNPs known for a particular gene, so that means there can be 2 to the power 5 combinations for these haplotypes. So these many different proteins can be synthesized, right? Now, if I'm talking about, this is the same thing that I was talking about. So if n SNPs are known in genes, there can be 2 to the power n possible haplotypes. And then which haplotype should be used for drug design and discovery, right? I just said that, okay, I am designing a drug against one mutated P53. But is that mutation common in the patients? What if it is only present in only 2% of the population? Then even if I design a drug against that particular variant, it might not be effective or it might not be that much useful, right? So we need to look into what are the frequency of these haplotypes and then design drugs accordingly. So one of the case study, right? So this tool simply uses a sequencing data and it gives you the possible haplotypes, and then you can choose the haplotypes you want to work with. So one of the examples that they have given, and it's really interesting, is this. They have talked about one monoclonal antibody which targets this receptor, FPR1, right? And this antibody is designed to reduce chronic pathogenic neutrophil inflammation. When the study was being done, then there are 5 non-synonymous SNPs reported for this particular receptor, right? Now, if I'm designing some drug against this receptor, then I have to actually, as I said, it's 2 to the power 5 different combinations of haplotypes. Then I should actually be testing the drugs for all the 32 different protein haplotypes. But that we generally do not do. We take the purified protein and we test it against, or we do some clinical analysis and we give the drug to the patients and see how it is affecting. And we know that in clinical trials, not all the people respond to the drug effectively, right? And it is because their protein haplotypes are different, right? So they have actually shown that just for one receptor, where 5 SNPs are known, non-synonymous SNPs are known, there can be 32 haplotypes of proteins, and the drug should be tested. If you're actually designing a drug which should be 100% effective, you need to make sure that the drug, in this case, your monoclonal antibody, it can target all the 32 different protein haplotypes, right? But that is really not possible. So what they have done, just to test whether this hypothesis is correct or not, this study was done in UK. Sorry, I'm forgetting the number of patients they sequenced. They took certain patients, right? They sequenced the patients for this particular haplotype. As you can see over here, this is the sequence data, and this is the data that they generated from their tool, right? So they sequenced and they saw that the type of variations, right? So these are the variants that I was showing you. So they saw that the frequency of occurrence of these variants was the highest. And as you can see, the trend, right? How the variations were different for different haplotypes. And similarly, they actually showed that there was an R square of 0.8 when they tried comparing the data from their tool and the actual sequencing. That means you don't really have to sequence the patients every time. You can use the make use of this tool to get an overall idea, right? Now, if you're designing a drug and you test it against these haplotypes, which are the most frequently occurring ones, then in that case, you are targeting the maximum subpopulation, right? So even if you do not test it against all the 32 haplotypes, if you can test it against the most frequently occurring haplotype, your drug, and if it works against those haplotypes, your drug can be a major success because most of the patients will be able to respond to that. Right? So this analysis, again, I have taken only a few sections from this study. If you're interested, this is the paper. You can actually have a look at it. It's a very interesting paper. It gives you an idea how big data, that is the sequencing data, it can be analyzed to get what is happening at the protein level. And then once you know what kind of proteins can be made, how you can calculate the frequency of occurrence of different protein haplotypes, and there can be different use cases of these protein haplotypes. I just gave you one example where it benefits you in making a decision for drug design and discovery, right? As a pharmaceutical company, I would not want to make a drug which is effective against only 10% of the patients, effective for only 10% of the population. I would want a drug which I can market to the maximum possible patients, right? So choosing the particular haplotypes for testing your drug can be one of the major applications over here, right? Okay. Now, again, jumping to a new topic, but related to big data, right? The more recent advancements in bioinformatics. Again, this is something related to artificial intelligence, machine learning, and AI, right? We talk a lot about these. So we know what generally a machine learning based pipeline is. You have a lot of data, as I said, right? So say I have data from, a lot of patients, and I know, okay, this drug, say I have 10,000 patients, I know among these 10,000 patients, these 5,000 patients, they were sensitive to a drug. These 5,000 people did not respond to the drug, right? But then all the patients, they have similar disorder. Why this drug did not work? I can actually extract patient data. For example, I can see what type of genes they are expressing, what type of proteins they are making, what type of mutations do they have in their genome, right? I can use all this data and I can see what are the factors responsible for resistance against a drug, right? Again, big data. I just collected data from the literature, or I generated my own big data and I'm using it to understand what is happening, and that can help me take an informed decision. In future, if I get a patient, I can see, okay, I found, okay, this ABC genes, if they are being expressed in very high amounts, this drug will not work. So I can test these new patients for expression of these three genes. And if there are high expression of genes, I know my patients will not respond to drug. I should look for another alternative, right? Again, only one example, but you can have multiple applications of AI ML, and very different domains of domains in which you can work with AI ML, and very different outcomes that you can expect. But the overall pipeline remains the same. You get data that you want to work with, whatever you want to understand it. You have to clean and process the data. As we say, garbage in, garbage out. So if the data is not high quality and clean, we cannot expect our models to perform good, or we cannot expect our models to learn from that data, right? And then we have the learning process. As I said, what factors, I said ABC gene, they are responsible for resistance. So that information is learned during this training process. And then I want to see, okay, my model said ABC3 genes, right? Is it really the correct answer? Then I take some data, independent data. I have not used it for training. And I know, okay, in this data, these 5 patients responded and these 5 did not. I put it back into my model and see, okay, the one who were resistant to the drug, they also have high expression of ABC. So I know, yes, the model, whatever it predicted was correct. And if at all there is any difference between the training and testing output, we can improve our own pipelines to make it more robust and effective, right? So this is the basic pipeline. But again, what data you want to work with is your choice. Cleaning is very important. And again, depending upon data cleaning and manipulation is different. What models or algorithms you can use to, train your model or extract information from your model. There are different algorithms available. You have to see what works good for your data. Then testing, we have different strategies for testing. And again, for improving also, we have different ways of feeding back the information. Okay, this is not working. We need to improve this aspect, and all can be done, right? Again, I'll give you one example, recent study, right? So as I said, we use data, we create an AI/ML based model, we do some predictions, right? Now, if I have new data, I can use the same model. This model can be used and I can predict the outcome, right? And if I know my model has a good accuracy, precision, and recall, then I can actually use this model to make, to have informed decisions, right? So again, one example here. So I was talking about similarity between small molecules. If you remember, when I talked about ligand-based drug designing, I said I can look for structures which are very, very similar, and I can have an hypothesis, okay, these are very similar, so they might do similar function or they might interact with the same protein, right? So this is one example here, this algorithm that we can use. There is an algorithm called Tanimoto or Tanimoto score, which tells me how similar two structures are, right? For example, this is my molecule 1, this is my molecule 2. I want to know how similar they are. We actually break them down into very small substructures. And this is like, if this substructure is present in molecule 1, this ring is present in both. So you have 1. This substructure, it is present in both. Yes, it is present in both. This substructure is present. No, it is not present. So we have a library of substructures and we see whether these are present or not. And then we use some formula over here, as you can see, that gives me a score, Tanimoto score. Okay, these two molecules are this much % similar. Right? So this is just one example. We have different ways of representing the similarity, right? We can have different fingerprints. What are fingerprints? When I'm talking about small molecules, these kind of structures, how do I explain what is the structure of this molecule, right? The 3D structure. To explain the 3D structure, we have different ways of representing it. We call them as fingerprints, right? I told you just one, Tanimoto, right? Then we have Morgan fingerprints, Feet Morgan, atom pair, RDK, torsion, layered, then, MACCS, MAC. So all these are different fingerprints that can help me understand the similarity between two different molecules, and they have different way of computing the similarity, right? Now we move to the actual example that I was talking about. So this group, what they did, they took 1410 FDA approved drugs, right? And we know when a drug is approved, in most of the cases, we already know what protein it targets, right? How it is performing the function it is being designed for, right? So these 1410 FDA approved drugs, they had 1262 known targets. Known targets means what proteins they were interacting with, right? This information we already had. So there were examples where one drug inhibited only one protein. There were examples where one drug inhibited 86 different proteins, right? No specificity. It's just inhibiting multiple proteins. So they had this data. These 1410 drugs, what proteins they are interacting with, they had a big table, right? So this was drug, and these are all the proteins which they are interacting with, right? Now, as I said, we assume that if two molecules have similar structure, they can do the same function, right? So if two molecules have very high Tanimoto scores and 90% similarity, I can say, okay, they can inhibit protein 1, right? So on the same hypothesis, what they did, they made all possible protein-protein pairs, right? So if I have 1410 small molecules, they generated these many drug pairs, all possible combination, drug 1 with drug 2, drug 1 with drug 3, right? You can see over here. And the fingerprints that I talked about, all these fingerprints, they used all the fingerprints and they calculated what was the similarity between these drugs, right? And they used these as features. And then they had one extra table that this drug 1 and drug 2, similarity I already know. Do they have any protein target in common? say drug 1 is also inhibiting P53, drug 2 is also inhibiting P53. So it has a common target, right? So in case they have a common target, they labeled it as a match. In case no common target, it is no match, right? So all the drug pairs, the similarity between those drug pairs calculated using different methods. And then the last column, which is your response variable, which says whether they have a common target or not. That means common protein that they're inhibiting or not, right? Not only similarity, they also had some molecular descriptors. What are molecular descriptors? They are again properties of these molecules, but in numeric values. For example, molecular weight of the drug, log P value of the drug, how many aromatic rings they have, or how many rotatable bonds do they have, all this kind of information. So they have almost 225 molecular descriptors. So along with this, so for example, this is your fingerprint, these are descriptors. So they had a big table, right? So the values in this table, they are describing your drug 1 and drug 2, and then you are telling whether they have a common target or not, right? So this was the training data that they used, right? So I talked about this algorithm. So I'm talking about this first stage, getting the data and preparing it. So this is how they prepared the data. So this is what I showed you. Now they built the model, training part. Again, training has a lot of different algorithms available. They used 3, logistic regression, naive Bayes, and random forest. They used all the 3 different algorithms and they saw which of these can best learn from this data, right? And they found out that random forest works the best, right? Now the model is built. So they use this data and they wanted to see if they can gain some information out of it. Now they wanted to see whether their model works good or not, right? So now this is one of the examples from the same study. What did they do? They took one database. It had a lot of molecules which are present in your food that we take, right? So they had many different compounds which are found in food, right? So as they prepared this drug-drug interaction data, now they had say around, I'm forgetting the number, maybe 600 something molecules that they extracted from the database. So it was like similarity of your first molecule, which they wanted to test, right? So it was like drug 1 with your food molecule 1. Then you had drug 1 with food molecule 2, right? So on up to, say, if they had 600 molecules, they had all these combinations with drug 1 itself. Then they started with drug 2, drug 3, drug 4. So they made all possible combinations. Again, they calculated all these values using different information. And what they wanted to predict was this. If I say that these 2 molecules are highly similar, as per my traditional hypothesis, I would say that they can inhibit the same protein, right? They wanted to see whether they can inhibit it or not, right? So when they did this analysis, making all these possible pairs, calculating similarity, and predicting the target, this is what they wanted to do, right? So based on these similarities, this is one small case study. So this molecule, they already had in their database, the 1410 FDA-approved drugs that they started with, this is one of those, right? And in their database, they knew that it interacts with all these 4 different targets, right? And one of these is COX-2. The common name is COX-2, COX-1, sorry. And this molecule, it showed high similarity with this, right? So this is another molecule that they found from that food database, right? Their model predicted that this trifusel, it is very structurally similar to this 5-methoxysalicylic acid, and it can also inhibit these proteins. So this is what the model predicted. Along with that, model also showed that this same molecule, it is also highly similar to this molecule, but it cannot inhibit these proteins. So what do you understand what I'm trying to say? Our traditional approach is if 2 molecules are highly similar, they can inhibit similar type of proteins or they have similar activity, right? But here we are trying to say it not always works. 2 molecules can be highly similar, but might not perform the same function, right? So this is an example of the same. So this is highly similar to, like, this is the molecule that they used for training. So this molecule showed very high similarity to both these molecules, but their model predicted that out of these 2, which are similar to this, only this can perform function similar to trifusel, right? And they tested this also. So as you can see over here, so this is a percentage inhibition activity, right? And this is against the only one of the proteins, which is COX, right? So out of these 4, they picked only one and they tested. So this is your actual molecule. They showed that it is like 42% of the COX was inhibited, right? Approximately. Then this is what they predicted that can inhibit COX, right? As you can see over here, it also had a comparable activity. Not exactly not as effective as your trifusel, but it also shows a lot of inhibition and it's comparable. But this molecule, which was similar, but was not predicted to be targeting this molecule, you can see over here, the inhibition potential is very less, right? So this is like one of the examples which show that there is a lot to learn from big data. They just collected a lot of data. They created a lot of drug pairs and they saw similarities. And then they added another information that whether they inhibit the same proteins or not. So there can be 2 molecules, very, very similar, only difference of one functional group, but the 2 can have very opposite activities. And this is the model that is helping you learn those differences between molecules, which make them different from each other. And it very well captures it also. So I just picked one example that they have proved, but they have actually shown the potential of their model in other cases also, right? So if you're interested in looking at this paper, sorry, I forgot to give the reference. I can share the reference. But this is again a very interesting paper which talks about the assessment of bioactivity using big data and machine learning based models, right? So this again emphasizes on the same, that big data for biological data analysis using artificial intelligence and machine learning based models can help you infer some intricate patterns which are difficult to be observed using your eyes or you know the manual analysis. And this accelerates the process and you can get results very quickly, right? Now, just to add to it, before I take a pause and we can discuss more whatever sections you like more. So this is another approach which is at trend, right? So when I showed you the different levels of hierarchy, I said we go from genome sequence, yeah, right? We find out what are the genes, what proteins it can make. Then it's not only what genes are present, whether they are expressing or not, right? So at individual levels, the expression of genes, proteins. Then I said, okay, when we talk about the expression, then what are the changes in the structures, right? If I'm talking about protein expression, whether it's the wild type protein or the mutant protein, then again the chemical biology, how they are interacting, the molecular mechanisms and all. And then we go to the system level, right? So right now what I was discussing, I'm either talking about one protein, 2 proteins interacting together, being inhibited by my small molecules. But in general, when I talk about a cell, I want to inhibit one cancer cell. It's not just one protein that I can inhibit. There are like so many different factors inside the cell. I need to talk about system biology of that cell, right? The cell as a system, and there are so many different players within that cell that are making it behave the way it is, you know? So it's not only one type of data, but we actually have to make use of multiple types of data. And as I said, because of the big data, all the data is available to me. When I talk about a cancer cell, I will not only talk about gene expression profiles. I can talk about gene expression profiles, protein expression profiles, mutational profiles, the metabolites they are making, the lipids they are making, the carbohydrates that are present, right? And then if at all there are some phenotypes which are relevant or associated with some other complexities, disease pathologies and all this information. And if at all any gene which is being a marker for the cancer, is there any drug available, drug response to which type of the variant of the protein? So a lot of information, and all the information is related to each other. So instead of using only one type of data, the trend is to put all the data together in the form of a network. And these are known as knowledge graphs. Whatever knowledge is available about a system, I can pool everything together and create a graph. And then I work on these graphs so that I also look into the associated knowledge, right? So for example, if I talk about any knowledge graph, biological knowledge graph, it can have information about the genes which are associated. Say this is a cancer-based knowledge graph. All the genes which are like the driver genes, passenger genes, how they're expression is different, right? Which of the mutations in the genes are lethal, right? When I'm talking about cancer, this disease becomes cancer, right? Then how is the anatomy changing? What are the cellular components that are being affected? What are the biological processes? If I'm saying gene, one is up, then what all biological processes it affects? If at all there are certain drugs which are designed for these diseases, what side effects these diseases can have? What is the pharmacological class of this disease? What is the molecular function of this gene? How is this gene regulated? What are the pathways it is involved in? And if I'm talking about disease, what are the symptoms? And these symptoms, which of these symptoms are targeted by the compounds, right? So this is again a very small example that even if I'm talking about one particular condition, there can be multiple sets of biological data controlling the actual effect that I see. So can I pool all this information together and then try and understand? So if you look into the system of knowledge graphs, again, this is also big data. It can be used to find out new connections and use the same, convert it into a machine learning based model. And the example that I showed you, right? There we had only similarity between molecules. I can actually pull in a lot of other information and it create a bigger model. So this can be created into either I can find new connections within this network, or I can create it into a machine learning based model and learn something from it and make new predictions, right? So one of the recent advances in bioinformatics at systems biology level is the work with knowledge graph, right? So I think with this, I will stop here. I talked about many different aspects from bioinformatics, starting from sequence to structure to drug repurposing to big data in bioinformatics, the systems level approach in bioinformatics, which is at trend right now. But then this is again very limited. Covering everything about bioinformatics in 2 hours again is not possible. Bioinformatics is very vast field, and again, depending upon what data you are working with, there are different ways in which we can analyze the data and make interpretations and take decisions, right? So with this, I will stop for now. And with this, I would like to acknowledge the organizers who gave me a chance to discuss about bioinformatics with the students. The institute I'm working with, IIIT Delhi. So everything that I showed you, it has been done at the institute itself with their support and resources. I'm also associated with the Center of Excellence in Healthcare, the health informatics part or the health related data. As I said, bioinformatics is not limited only to health or human data. You can imagine the analysis on sequences, proteins, if I'm talking about plants, right? The various aspects of plants. I want to improve the quality of plants. I want to make plants, adapt to the changing environment, or many other things. The different diseases that plants can encounter and everything. All those studies can be done using the same tools, but the data will be different. We start with the plant data, right? Or animal biotechnology, animal bioinformatics, right? You can do everything for the animals as well. And some of the works that I showed you, they are from the current research projects in my lab. And I am thankful for the funding agencies who are supporting that work again. And some of the results that I showed you, they are in collaboration with one of the institutes in Japan, AIST, our hospital partner, which is Mex Healthcare, and IIIT Delhi, where we work for in vivo models, right? And this is my webpage. You can actually look into the other research projects which are going on, the publications where we have published all the available literature. And if at all you need any guidance, any support in any terms, you can always reach out to me at this email ID, and I'll be happy to help you. So with this, I will stop. And I would like to ask the audience if they have any questions that I can answer, or any topic they're interested in and they would like me to talk more about.
Okay, thank you very much, Dr. Jasper, for giving such an excellent presentation. Wow, I got a lot of new insight by listen your lecture today. Okay, so now we have come to the Q&A session. To all the participants, you can send the raise hand emoticon first, maybe, and I will let you to deliver your question directly to Dr. Jaspreet, or you can write your question using chat box features. So, and don't worry, you can deliver your question using English or Bahasa Indonesia. I will try to translate your questions then. So I will open the first termin for maybe please participant, you can raise your hand first and I will let you to deliver your question. Silahkan peserta yang hadir boleh untuk raise hand and I will let you to deliver your questions. Okay, we already have one participant in here. Bu Fitria. Bu Fitria is one of our lecturer, Dr. Jaspreet. So maybe Bu Fitria, you can speak, yeah, deliver your question directly to Dr. Jaspreet, please, Bu Fitria.
Okay, thank you, Miss Daniel.
Sorry, your suaranya agak kecil, Bu Fitri.
Oh, oke. Halo.
Okay, thank you.
Suara saya
Yes.
terdengar, Bu Daniel? Sudah jelas?
Yes, clearly enough. Thank you.
Clear. Okay. Okay, good morning. Thank you for Miss Daniel. Morning, Dr. Jaspreet. It was so interesting that bioinformatics research has expanded into human genome research now. Yeah, so then your presentation shows how bioinformatics has become a critical tool in dealing with the pandemic. Yeah. And then influencing everything from basic research to public policy decision making, maybe. And then I think this is turn has a direct impact on people's daily lives, from the ability of accurate diagnostic tests and then to the rapid development of vaccines, to the implementation or more target public health policies. Yeah. And my question is the first one, how do bioinformatic tools help in predicting potential zoonotic transmission of SARS-CoV viruses, maybe, for example? And then the second one is what role does bioinformatics play in predicting and preparing for the future coronavirus outbreaks or maybe for another disease outbreak? That's all, I think. Thank you for Dr. Jaspreet and thank you for Miss Daniel.
Okay, thank you so much, Bu Fitria. Jason, maybe you want to answer the question or we... Wait, okay,
I can
you
answer, yes.
want to answer. Okay.
So yeah, thank you for your question, Dr. Fitria. So as I said, like I only discussed only a few of the studies which I thought might be good to begin with or to grasp what we can do with bioinformatics. But as you said, the transmission aspect itself was very important during this analysis. And there are papers, if you want, I can share the references where people have shown that why SARS-CoV-2 was more severe in comparison to the previous strains, which which they observed in pandemic. So when they did this sequence analysis, even using these, so we know that when we talk about this TMP, RSS2, and ACE2, it's the spike protein of the virus that interacts with this, right? So they have done this analysis. They have seen what are the interactions between these receptors on the human cells and the residues in the spike protein of the virus that interact. So when they did this analysis, they have realized that SARS-CoV-2 had a lot of variations in its spike protein, which increased the interaction affinity between the human receptors and the spike protein, because of which even if it interacts for a few a fraction of seconds with the cell, the interaction is strong enough to keep it bound to that. So that's why the transmission rate was higher, or the rate at which it was getting into the humans for infectivity, that was higher in comparison to the other variants. So this is like one of the ways in which we can do. Then there are a lot of structural studies also which show the exact molecular mechanism, how it is infecting. Then if you're talking about like which of the hosts, as you talked about if you are talking about only the transmission to the human race, or if you are talking about what all animals they can use as hosts. So this is one of the studies that I can think of as an example. And sorry, I forgot about your second question. If you can repeat or give me a little hint about that.
The second question, yeah.
The second question is, what role does bioinformatics play in predicting and preparing for the future
Okay. Yes.
another disease outbreak?
Yes. So this is like one example, the interesting example that I was talking about, though this is very old study. I hope you can still see my slides. This is an old study from 2009 itself. So this work is from Google itself. So if we talk about ourselves, even if we have some slight symptoms, we generally try and look into Google to find out what could be the diagnosis for ourselves, right? That's the first thing that we generally do these days. Everything is available. So just by the queries that we add into Google, right, they actually developed a method that can help predict when will be the next viral outbreak. Right. So if you know, many of the countries, they have vaccination drives for, the virus is like a seasonal kind of a thing that for them, influenza virus, and they have vaccine drives for those, right? So just to predict when will be the next wave, or when is the next wave coming, and which of the areas have started getting the infection outbreak, right? So I'll just show you one example over here. So they just use the data query. So this there is a health organization called CDC. So they generate these reports every year for the influenza infections. So how many cases, as and when they report, to the doctors, weekly reports. Okay, these many patients came and everything. So they used the previous year's reports. So they took reports, I think 2007 to 8, and during that time, what were the queries people entered into their search engine? So these are some of the queries that people added. Multiple queries, they actually grouped them. For example, people were searching for the complications associated with influenza. Then what are the home remedies for cough and cold? Then what are the general symptoms? So these are the general queries that we also generally put. So they clubbed them into different groups, and they used these queries to see whether an area is experiencing an outbreak in the influenza or not. And then they used these queries as a training data set. They modeled their system, and they also validated it with the reports, the CDC reports, which were coming out after that point of time. And their model was, as you can see over here, right? The black line is what their model has estimated, how the trend is increasing, and the red line is the report from CDC. So there is almost a close overlap between the same. So if we use these queries, we can actually predict beforehand that there is an outsurge in the number of influenza patients. And that too, if we can do this analysis on the basis of different areas, this area is suddenly experiencing an outsurge in the influenza patients. So you can devote more resources to that area instead of the others. More vaccinations are to be given in that area. That area needs to be prioritized. So this is like one example. Again, this is very old study. If you look at the recent trends in prediction of disorders, the models are highly effective, and we can use them on day-to-day basis. So not only this, so this is another study. This is also from one of my colleagues in IIIT Delhi. He very closely works with clinical data. So AIIMS is like one of the biggest hospital in India, chain of hospitals. So he's working to predict thermal shock in babies. So the babies, like the few initial years for the babies are very critical. So just by looking at the images of the baby, they are trying to predict whether the baby will get into septic shock or not. And they can actually predict this in almost 8 hours advance. So this is again a earlier report, but they have refined this model, and they have actually deployed it in the hospitals. The doctors are using it these days, and they are trying to right now they're not making any clinical decisions based on it, but they are trying to see how effective a model predicts. If the model predicts, okay, this baby might get into a shock and these are the chances, the doctors observe the baby more so that they can avoid it. And how many times the model is actually predicting it the correct, it is giving the correct information, and certain measures are to be taken immediately to protect the baby. Otherwise, the baby won't be able to survive. And if we can predict this chance of getting into the septic shock 8 hours ahead of the onset of the shock, then we can actually save a lot of lives. So this is one of the, again, practical examples where image data is being used, and there are models which are training on this image data, and they are helping us to predict the outcome of any health related decision. So like this, there are multiple cases. I just had a few to share. Yeah. I hope that answers your question.
Thank you. It's so clear. Thank you.
Okay, thank you, Bu Fitria, for the question. Do you still have a question more, maybe, or it's quite enough?
Quite enough.
Quite enough,
Okay,
right?
thank you.
Thank you. Okay, I will let other participants. You can raise your hand, and I will let you to deliver your question, please. Okay, we have Bu Anissa Wulan in here. So, Bu Anissa, you can maybe open your mic and.
Okay, thank you, Miss Daniar.
Yes.
Hello, Dr. Jaspreet. Thank you for the very interesting information. I would like to ask, what are the common challenges researchers face when using molecular docking, and how can they be addressed? And what are the key factors that influence the accuracy of molecular docking simulation for in your research? Thank you, Ms. Daniar.
Okay, thank you. It's clear enough for the question, Dr. Jaspreet?
Yes. So I'll just
Okay.
repeat your question in case there is anything else. You can just tell me again. So you're asking what
Yes.
are the challenges or in using molecular dynamics, like molecular docking and molecular dynamic simulations, and what are the key factors that we should take into consideration, and the accuracy depends upon what factors, right?
Yeah.
Yes. So the key challenges, again, when I talk about molecular dynamic simulations, as I said, protein, if I just keep the protein static, right? First of all, we are taking the structures of the protein from PDB. The protein, when they are trying to crystallize it, it's not always in its native state. To make a protein crystallize, or to crystallize a protein, we have to do a lot of changes in the protein, or we have to put the protein under certain conditions so that it can crystallize and we can solve the structure, right? So first thing is, the structure that we get from PDB, it's not always the native structure, right? It has undergone some changes that are not present in its normal physiological conditions. So whenever you download a structure from PDB, you need to relax the structure using, again, some algorithms or a small simulation technique so that it can attain a more relaxed structure. That is the first thing. Then when I'm talking about docking, generally what we use, because of the resources, we generally use rigid docking, where the protein also is rigid and the molecule, that small molecule that you are taking, that is also rigid, right? And you are trying to dock. Again, the protein is not rigid in the body. There, say this is my active cavity and I'm trying to fit a molecule, but in actual, this cavity keeps on moving, right? So there can be chances that the ligand, if not fitting it like this, if the protein moves a little, it can fit in, right? But now we have advanced algorithms. For example, we have semi-flexible docking, we have flexible docking, where if I know this is the entire protein and only this much is the active cavity, I can make the residues flexible only in this. But then the computation time increases. You need more resources for those. Again, the ligand also, wherever the rotatable bonds are, the ligand can take different conformations. So we have to generate all the conformations of the ligand. We have to see what are the residues of the protein interacting with the ligand. We have to make them flexible and then do the docking. And then a lot of poses get generated. If I'm saying this protein can take 5 different conformations, this ligand can take 10 different conformations, so 5 into 10. All those conformations I need to study. So the sample space of these protein ligand conformations is huge. So algorithms that can sample everything, again, they are complex. So do you require better resources if you really want to do very accurate docking and simulations, right? Then once the docking is done, you come to molecular dynamic simulations. Again, for how long are you simulating that structure, right? So if you are talking about just looking at the stability, whether it stays in or not, people generally go for 100 nanoseconds, 200 nanoseconds, 500 nanoseconds. That is good enough. If the ligand stays in for that long, we can say that it will, there are less chances that it will just pop out during longer simulations. But if you are looking at conformational changes in the protein, you are talking about, you are looking at the transporter which opens and closes in a membrane so that a small molecule can pass. If you're looking at these kind of conformations, you cannot expect this to happen in 500 or 1,000 nanosecond scales. You have to really run longer simulations, and even with good computational resources can take weeks to months. So this is like one of the challenge. So if you have good resources, you have good GPU cards, you can do calculations quickly, you can finish these simulations in smaller time as well, shorter time as well. But still, resources generally are the limitation. And then the force fields, like at the back end, whatever calculations you are doing, the parameters that you are using for that. So if you're taking some small molecule which has some atoms which have not been characterized, like for which the parameters have not been characterized, so you might not get accurate results. So you will have to work for parameter generation for that particular atom and then work. But still, for most of the heteroatoms and for the proteins, we already have good force fields. So which force field to choose for the calculations, which software to use, then what time steps, again, the parameters in the simulations. Just to run the simulation faster, we might increase the time steps, but then the accuracy of the result that you get, it decreases. So more computational resources definitely for molecular dynamic simulations are still needed, and good amount of time and good analysis of the data that you're getting. Just taking the last frame, generally what people do is they simulate the structure and they take the last frame, which might not be the correct idea. So you need to, sample the conformations that the protein has taken over time. And then from there, you need to choose the one which stayed for longest, or, where the conformations were the highest. So that could be one of the stable states for the protein. So again, this is not just a technical protocol that you just run and finish. Depending upon what you are studying, what kind of conformational changes you want to observe, you need to decide which algorithm, what time frame you want to choose, and how do you want to analyze your data. Does that answer your question?
Okay, thank you, Doctor. Thank you, Ms. Dania.
Okay, thank you, Bu Anissa, for your thoughtful questions. Yeah, we still have a time for maybe a couple of questions, several question more. So who will like to be next participant? Maybe from student, please. If you have any questions, you can deliver with Bahasa Indonesia, and I will try to uh, translate your question. From student, maybe? Any question? Okay. We have a question from student in chat box, but it's written with Bahasa Indonesia, so maybe I will try to translate the question first, Doctor. Good morning, all lecturer and Dr. Jaspreet. My name is Niputu Laksmi. I want to asking the question to Dr. Jaspreet. Bioinformatics is one of the subject that teach in the high school in Indonesia, or especially in Indonesia. Sorry. Okay. After she learned your lecture, she think that bioinformatics must be teach earlier in the school, for students can be understand more about the role of bioinformatics in their daily life. But and we also already know about NCBI. But maybe can you maybe introduce us the kind of NCBI website that maybe more simply to introduce to our student in high school level or maybe in the junior high school level, so we as a teacher can more can introduce the bioinformatics from earlier, maybe something like that. Dr. Jaspreet, the questions. Thank you.
Okay. Thank you for the question. So I was really amazed. So as I said, I was in Indonesia a few weeks back. So I was
Okay.
one of the speakers for a conference in University of Bravegia, and I also interacted with
Okay.
some researchers in Brin. So actually, I came back on October 1st only. So and
Okay.
yes, and there was a competition for high school students to present their research, and I was really
Yes.
amazed the way students
Oh,
presented the research work.
okay.
The understanding of bioinformatics they had, the tools that they were using and presenting, it was really nice. So I'm really happy that you people learn bioinformatics in high school. We still do not have it over here. People start
Okay.
learning it only in specialization once they get into the college, right?
Okay.
So if you are doing it in high school, really, again, because bioinformatics now is an inseparable part of biological research, you cannot do research without understanding bioinformatics.
Mm-hmm.
So if you're learning it at this stage, it's really good. And again, if you're talking about an alternative to NCBI, I would say NCBI is one of the biggest resources available. So it will
Yes.
be good to get acquainted to NCBI for sure. Yes, but
Mm-hmm.
not maybe all the databases. We can pick a few. For example, for genes, we can talk about GenBank, right? For protein, personally, I like using UniProt more. It has
Mm-hmm.
almost every information in one place that you want, and it is also linked to external resources. So if we talk about, biomolecules, and definitely you cannot start even before, like much earlier than that. But when we are introducing students to what are proteins, what is DNA, probably at that point itself, we can talk about the sequence of DNA, sequence of proteins, where we can get these sequences from. For example, when I'm talking about gene sequence, we can get it from, GenBank. Then when I'm talking about protein sequence, I can get it from UniProt. We can introduce them to the sequence analysis. For example, BLAST. Almost everyone uses BLAST now, right? So it has become one of the primary requisites for any research when we are talking about sequence analysis. So we can introduce them to BLAST. So that will also help them understand some of the statistics behind these tools, right? So when we are talking of BLAST, similarity, identity, e-value, I think that will help them develop curiosity. And with time, when we are introducing structures, we can show them, we can introduce them to, again, protein data bank. We can download a protein structure, whatever we are giving as an example in the class, we can download that structure. We can use some visualizing platforms, show them how a protein, we see a protein, like we have not seen proteins as such, but how we see a protein looks like, so that will help them engage. So just reading that there is protein, there is DNA, but not getting to see it probably is one of the limitations. But if we can show them in using in silico methods, I think that will really generate curiosity in the students. But overall, I'm really happy with the knowledge that the high school students have and their inclination towards the research interest.
Okay.
So I think you're already doing good. You can just advance it a little so that you keep on updating yourself with the advanced topics. And as you rightly said, that introducing bioinformatics at the earliest stage would be the best solution for the students. So as I said, as and when you introduce what is DNA, what is protein, we get them introduced to the databases, tell them how do they access it and how do they use them for very preliminary analysis, multiple sequence and alignment, pairwise alignment. That itself will be good.
Okay. Thank you so much, Dr. Jasper. How about Niputu? Sudah terjawab pertanyaannya? Do you
Yes,
still
Bu.
have? Sorry?
Terjawab, Ibu, terjawab.
Okay, thank you. Okay, thank you for your good questions. Okay, we still have a time, so please to other participant, maybe you still have a curious thing to, um, sorry, to ask to the our speaker for today.
So if you have any questions
Okay.
apart from this, you feel free to ask. It doesn't really have to be related only to the slides, because as I said, I could not have covered, I already feel that I've covered too much. It could be overwhelming, but,
Okay.
I've already covered a
Mm-hmm.
lot of aspects for the people who might not have very good hold of mathematics. But still,
Okay.
if you have any question beyond the slides, the topics that I discussed, I'll be happy to take those as well.
Okay.
And I'll
Thank you.
try to answer as much as possible.
Thank you, Doctor. But maybe you can share your slide for today materials to us. Maybe
Sure, I
later
can
I
do
will
that.
email you about the
Sure, I
share
will do
your
that.
slide. Okay. Okay. So still have a question from participants, maybe? Ada pertanyaan lagi, teman-teman? Boleh you can deliver with bahasa Indonesia, I think. I will try to translate. Masih ada pertanyaan, teman-teman mahasiswa silakan boleh di kolom chat atau bisa raise hand. Okay, maybe waiting the question from students. I'm really curious about your research in jamu, doctor. You already said jamu, yeah. We Indonesia have a traditional medicines, as you already know, maybe jamu and what we call ashwagandha.
Ashwagandha, yes.
Ashwagandha is one of the kind
Yeah.
of traditional medicine from India, but maybe we don't have that in here. Maybe if you already have a research about traditional medicine, especially doing collaboration with Indonesia researcher, maybe do you want to you can to sorry, would you like to share maybe with us about the update
So I do
information
not
maybe?
have any collaboration at present, but I'm looking forward
Okay,
to it. So as I
okay,
said, this was my first visit to Indonesia, and I had I had a chance to discuss a lot of work with many different groups. So
okay.
probably in the coming year, I'll have something to show on papers with collaboration with Indonesia. But yes, I got to know
Okay.
about Jamu there only. So there were a few faculties who talked about their work in traditional medicinal systems and
Mm-hmm.
how the two systems are similar also. So ashwagandha is like
Mm-hmm.
only one of the plants that we very commonly use in in
Mm-hmm.
Ayurveda. So
Okay.
it is used the root powder of ashwagandha is very commonly used, and it is available in the market also to consume.
Mm-hmm.
But then as my work with related to ashwagandha, we have shown that it's not only roots, even the other
Mm-hmm.
parts of ashwagandha are equally important, and depending
Okay.
upon what type of activity you are talking about, right? So if I talk about role of like the usefulness of ashwagandha for insomnia or brain
Okay.
health, we can talk about roots. But when we talk about anti-cancer activity of ashwagandha, it's mainly the leaves which are important, because the component I showed you two components, vidafrin and vidinone. So, and for
Oh.
anti-cancer activity, vidofrin A is more important, and it has more concentration in leaves. So,
Mm.
similar kind of works that we have discussed, that they
Mm-hmm.
are also looking at different extracts from different parts of the medicinal plants that they use, and they would want to categorize like what are the bioactive compounds in the extracts. And if I'm saying this extract is helping in neuroprotective activity, then what are the molecular mechanisms behind it? So we would
Mm.
want to work together. So I did not show you my work using the transcriptome analysis. So we do a lot of transcriptome analysis. For example, if I already know that from like some clues in the literature, I already know that ashwagandha, let's not talk about ashwagandha, let's take some other molecules. So I said that we are working with black lily, tectoriginin. So if
Mm-hmm.
I have some clue that it has neuroprotective activity. So we take
Mm-hmm.
certain cells which can differentiate into neurons, and then we treat it with different doses of tectoriginin. We extract all the RNA from the cells, and then we sequence the RNA, and then we see how these tectorogen treated cells are expressing different genes at different concentrations in comparison to the control. And using these differences in the gene profiles, we try and find out what could be the molecular mechanism of action. So these kind of studies are something that we are planning in collaboration with the University of Pravijia. So hopefully soon it will keep off and we will have something together.
Okay, thank you. Actually, one of our lecture in our department, we have one lecture that have a research in traditional medicines, jamu. Ma'am Ayu, but maybe for today she can join with us in this Zoom link. So I will try to maybe tell her about your yes perspective collaboration in the future. Okay, thank you,
Sure.
Dr. Jaspreet. Okay, we back to the question and answer session. Maybe to all participants, do you still have any questions? We still have maybe time 5 minutes. Okay, Mr. Rizky already raised hand. Maybe you can open mic and deliver your question
Yeah,
to our speaker. Thank you.
thank you, Mr. Daniel. I do apologize. I cannot turn on the camera because of the
No
signal.
problem.
I
No
hope it's
problem.
okay. Yeah.
Yes, yes.
Thank you so much, Dr. Jaspreet, for your explanations for this today lecture. Related to the bioinformatics, my name is Rizky. I'm from the biology department. I'm also teach bioinformatics to students, and I just want to ask and curious about uh, telling bioinformatics to students, especially sometimes needs how you call it, like logics or basics core that can make, let's say, the simulations of the protein. Sometimes we do the simulations and making the modifications of this part and to that part and so on. So that's why we found that the ligands and then the site that are suitable with the uh, purpose of our research. Those modifications needs good logics and basic course of biology, especially of proteins and also some genes maybe to get the purpose of our research. But sometimes students just try and see the results. So it means they just know they didn't know the
Process.
purpose of the modifications and then the how why we made like this, why we changed the conformations, why we put like that. Maybe you have a tricks or like how you call it, advice, how you get to know the in bioinformatics or tell the bioinformatics to the peoples so that the modifications or the bioinformatics process that we done is can be easily and more effective, and they can understand easily why we do this part, why we have to do that part, why we have to modify this. So the purpose of the research will be reached, because sometimes how you call it, the most difficult of doing the bioinformatics, we know this is just a simulation. Sometimes we do trials and errors and something, but if we already know the core of the biology or the mechanism, I think it will be more effective and more efficient of the process. Maybe you have advice or tricks how to explain that. Thank you. Before.
Yes. I can try to answer it, but as I said, we do not have a lot of exposure to high school students for bioinformatics. We do teach them it can be just one chapter in the entire course, but not very extensive, because definitely you can use bioinformatics only when you understand biology, right? These are just tools to infer biology. But if you do not know what proteins are, what is DNA, then it is difficult to make them understand. But if you have already, like the students already know this much biology, like what is cell, what are the different biomolecules in cells, and what is the function of those, and you are doing that, like you are taking it to next level, you are expanding it a little and introducing, as you said, what are mutations, then how you can study mutations, how protein changes upon mutations. If you're trying to introduce this, if I would be in your place, then I would start with a story, a case study. For example, I introduce them as like I will just hypothetically make one patient, and I would say, okay, this is one patient. This patient has gone to doctor with these symptoms. Doctor has diagnosed the patient with, say, as I said, say I'm talking about the Wilson disease. So I say, okay, patient has been diagnosed with this Wilson disease. These were the symptoms, and looking into the DNA of this patient, say probably we did not know what is causing this, if it is new symptom, and if Wilson, I already know what could be the possible cause. So I look into the molecular mechanism. I can show, okay, this is the DNA sequence from normal person, and this is the DNA sequence with this change in the diseased patient. And then I can actually show any, even if it is a hypothetical structure, I can say, okay, for the normal people, this DNA, it can make this particular protein which gets into the membrane of the cells. And for this patient, this protein is getting formed, but see, here is the structural defect. And because of the structural defect, even if it is there in the membrane, it cannot open or close for the exit of the copper. And then from there, we can introduce, okay, see, this structural defect, it is because their DNA had some problem. So this DNA could translate into this kind of, amino, like instead of this amino acid, it got translated into this amino acid. And if I say not only talk about this patient, I bring 5 different patients, they will have different types of changes in the amino acids, but each of this is resulting in the structure distortion. And then how these changes in the structure are affecting. So not every change in the protein will result in the same type of effect, right? Some might not let protein open, or some changes are leaving it open for very long. We don't want this long. And for some mutations, the protein itself is not forming. So I can give examples of these. And then, okay, if I want to study these proteins, how can I go about? So first thing is I just compare the sequences. So I tell them how the sequence comparison can help there. And then I can tell them, okay, proteins don't function till they fold. So how do I compare the structure of the protein? So I can show them, superimpositions of protein in pi mol, and where are the regions which are different. So instead of only theoretically teaching, if I can show them how the structure is changing in with different mutations, I superimpose them in the pi mol. They can zoom in the structures. They see, okay, this is only minor defect, but still it is making huge impact. So then if we say, okay, say I just found out that there is a new mutation and it does not, I do not have a structure for it already. So how do I make a structure? I can take a wild type protein. I can use again some in silico tool, mutate it, and then say, okay, mutation has happened, but this protein has to change. How we study these changes, we can introduce molecular dynamic simulations, help them understand a little, but then molecular dynamic simulations is a higher concept for high school students. I would not suggest getting too much into the depth of those, because till then they don't even understand the physics rules that we talk about at the back end and all. So probably they will not be able to connect to what we are talking. So if we can just teach them about the sequence analysis, structure analysis, the different tools that work up to the structure part, if the structure is already available. And even if not, then we can teach them about the modeling of the structure. So these things we can easily cover at the high school level also. And then if there is some student who is interested in learning more, then we can introduce the higher concepts to them.
Okay, thank you so much, Dr. Jaspreet. It's a very great answer. So the case-based learning is so important in this bioinformatics. So we give a lot of case first, and then before the hands-on of the tools or the website or the software that we will use. Thank you so much for the answer.
Thank you.
Okay, thank you, Pariski, for the questions, and Dr. Jessel for the answer. Okay, I will ask again to all the participants. Do you still have questions, maybe? Okay. Okay. Yeah. Yeah. I think they don't have any question again, Dr. Jaspreet. So because we have a limit of time too, we need to end our discussion session for today. Thank you very much for participating in Q&A session to all the participants, and thank you so much to Dr. Jaspreet to answer all the questions with a very amazing answer. And yeah, we have finally come to the end part of this sessions. To Dr. Jaspreet, are there any final thoughts before we end this session? Maybe like a closing remarks to all of us in here. Maybe you have something to say to closing your presentation.
Oh, so you're asking me for the closing remarks?
Yeah, for the
Okay, sorry.
from you,
Yeah.
closing remark from you. Maybe you still have something to ask, yeah, about about the bioinformatics itself to share with me with us in here.
So I would just say, as I already mentioned, what I presented is only a small part of bioinformatics. Bioinformatics is huge. It can have potential applications. Even though the resources or the softwares are the same, how you implement to answer your research question, how you use your creativity and innovation to answer something, So there can be one tool for sequence analysis, but people have never used in one of the ways. You think about it, you come up with using it in your research, and you can come up with very innovative answers to the problems which are out there. So, the scope is unlimited, and your contribution in the research field can be one of the turning points, right? So I would really suggest all the biology students to start getting themselves equipped with the bioinformatics, and not only tools and softwares. I think because with the trend of big data analysis, training yourselves to understand big data and make use of tools and concepts of statistics, machine learning. So if you can start learning these as well as you move forward, I think if you start early, then you can actually do a lot of good work with the data available. And it's not just that you have to produce data in your lab every time. And as I mentioned, there are a lot of resources available online. People generate a lot of data to answer one specific question. I might have one very specific research question, but the data that I'm generating, it can have answers to many, many other questions. So whenever you have some question in mind, just look into the open repositories. Do you have data available to answer that question already, instead of generating new data? So if you already have data available, you can use that and you can answer that. You can frame questions, methodology, and you can do the analysis. And if you do not have data, definitely like use your experimental skills and generate new data. But then with the data out there are many unanswered questions that can be looked into and effectively answered. So so much to learn, but then the time is the limiting point. So don't wait for you to graduate, pick up some problem in your PhD, learn. Just keep on doing
Okay.
small courses online, and that will help you go very far off.
Okay, thank you for the concise closing remark, Dr. Jaspreet. Now, please allow me also to take some maybe several highlight points from your presentation, this session. The first one, maybe we have to kind of what we call, there are sequence-based information and structure-based information. So you already explained a lot about the proteins and genes, some things like that. And after that, bioinformatics itself is about a compile of many biological data and need one, and you said that this big data need one platform to save or to documentation all the information. So you already tell us there are many platforms that we can use as what we source of data like NCBI or PDB, protein data bank. And I really get into the information about SARS-CoV-2. Like you said, 85% genome from SARS-CoV-2 is similar with a bat genome, but have maybe several mutations, as you already said. And bioinformatics also help us to find the similar protein or genes in database for making drugs. For example, when the SARS-CoV-2 happened in this world, we already know uh, the virus has RDRP. Sorry if I can clearly the proteins, and we already know there is a drug can against this protein, and so we use that data to develop a new drug to against SARS-CoV-2. And ashwagandha is one of the your research maybe, yeah. We know maybe Indonesia we call it ginseng India, ginseng India. But we in Indonesia don't have ashwagandha, but we have a similar plant like ashwagandha, and we already have a traditional medicine. So like you already know, it's called jamu, and jamu have so many ingredients in it. Like maybe one of them is we have temulawak, jahe, and ginger itself. And at the end, in drug design, maybe one type of gene can be expressed to variety kind of proteins. That means the same drug can be have so many variety response in each person. So you need to do experiments, yeah, Dr. Jasper, to find the really right one of the drug uh, uh, based on drug design that we maybe made in our research. Okay, maybe that's several highlights that I can that I can make to conclude our discussion today. And finally, I would like to thank you again to Dr. Jaspreet for joining us in Biology Education Visiting Lecture 2024 and delivering an interesting and insightful, very new thing for maybe for us by your presentation. And to all participant, ladies and gentlemen, please give applause maybe to our speaker once more. And also thank you for all participants for the participation in the sessions. And yeah, before I ending this session, maybe I want to remind you, don't forget for our upcoming event next week, to all the participants, we still have the session 2 with Professor Lijan Lester from Sam Houston State University, United States of America. She will give an interesting topic about TPA-CK for biology teachers. So please stay tuned and have a wonderful day. Maybe I will end this session for today. Thank you again. Wassalamualaikum warahmatullahi wabarakatuh. Good afternoon, and please, MC, you can take over the next agenda. Thank you, Dr. Jaspreet.
Thank you. Thank you so much.
Thank you. Silakan dikembalikan kepada MC.
Okay. Thank you. Thank you, Dr. Jaspreet or Dhanjal, for your time and expertise today. The session has been both enriching and insightful, shedding light on the role of bioinformatics in education. We also extend our gratitude to our moderator for skillfully guiding the discussion, Ms. Dania. I'll remind again that participants are expected to fill in the attendance form using the link provided in the room chat. We are excited to continue our October series of back-to-back general lecture and visiting lecture. I'll remind again the Ms. Daniel said, but next week we will welcome Prof. Deacon Lester from Sam Houston State University, USA, who will be presenting on TPA-CK atau Teknologi Pedagogical Content Knowledge for Biology Teachers. The session is part of a continuous agenda throughout October, focusing on diverse topics to enrich our understanding and skill in the field of education. Look forward to your participation. As we approach the end of today's visiting lecture, I would like to thank all participants, invited guests, and faculty members from the Biology Education Department for your presence. May this event bring benefit to us With this, I, Dini Rahmawati, your Master of Ceremonies, bid farewell. Please accept my apologies for any shortcomings during the event. Good afternoon. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you to all participants. Thank you, Dr. Jaspreet. I hope can see you in another occasion. Thank you.
Sure, sure. Thank you so much. Thank you, everyone. Thank you.
Thank you. Terima kasih Bapak Ibu yang sudah hadir. Terima kasih teman-teman mahasiswa. Saya izin